Good morning, good afternoon, and good evening, depending where you are. Welcome to the 2022 Sentinel Innovation Day. It's a great pleasure to have you join us via Zoom. And my name is Sebastian Schneeweiss. I'm the principal investigator of the Sentinel Innovation Center. And I'm going to kick us off today for a day of exciting updates on a bunch of projects. And with that, I, of course, want to acknowledge the 4 key partners in the Sentinel Innovation Center. That is Duke University, Vanderbilt University, Kaiser Permanente in Washington, and the Brigham and Women's Hospital. And you will hear from investigators from all of those 4 groups throughout this day. Next slide, please. The agenda today is just brief welcome. Dr. Robert Ball from the FDA will provide some opening remarks. Then we hear from 7 projects that are ongoing. Some are more progressed than others as part of the portfolio of the Sentinel Innovation Center, and then some closing remarks. Next slide, please. Next slide. As just to set the stage, I think it's important to talk briefly about Sentinel as a whole. Sentinel builds on nearly 1.5 decades of success. Together, we have created a sophisticated, robust, privacy-preserving system that uses quality-checked and curated information covering a large fraction of the US population to provide regulatory-grade information to the FDA and others. And in addition to performing its primary mission of providing information about the safety of medical products, Sentinel has increasingly become an important resource for FDA's real-world evidence initiatives. It also demonstrated its value on important public health surveillance programs during the COVID-19 pandemic. And Sentinel has also proven to be a national resource for an array of public and private sponsors, and it has become a model for national and international programs around the globe. Now, from day one, and I was involved from day one in Sentinel, Sentinel had a strong focus on developing advanced methodologies for data processing, data cleaning, and data analytics that make research on the safety of medical products scalable, but also their results actionable. And since late 2019, these projects, these methods-focused projects, were bundled into the Sentinel Innovation Center that I'm heading for the next for 5 years. Next slide, please. If you click another time, please. The Sentinel Innovation Center's main focus is to do research into data processing and data analytic methods that include electronic health record data, including the structured, semi-structured, unstructured information, in order to expand the data analytic reach of the existing Sentinel infrastructure. So the Innovation Center works very closely with the Sentinel Operations Center. We're highly integrated in doing research that complements the already existing data infrastructure, analytic infrastructure, and will ultimately expand that structure. We have 4 initiatives in order to achieve that. One is focused on, and you will hear from all examples from all 4 initiatives. One is the Data Infrastructure Initiative, the Feature Engineering Initiative, where we extract information out of these types of electronic health record data, causal inference based on these types of data, and detection analytics. How can we more efficiently detect signals that we did not otherwise expect? Next click, please. Ultimately, can you go 1 click back, please? Ultimately, the goal is after 5 years to develop a network of covering at least 10 million subjects, where we have not only the EHR data available, but we also have the linked claims data information available. We always call this the best of both worlds in a way that can be queried by the Sentinel system in order to expand the reach of the already existing Sentinel infrastructure. Next click, please. Here you see an overview. This is all public information. You can download this from the Sentinel webpage. The strategic plan of the Innovation Center. You see the 4 initiatives highlighted on the left side and the various programs that are interconnected and building on each other in order to deliver this vision of 10 million lives that we understand and can query in a meaningful way in order to understand the safety of medical products. Next click, please. So yeah, 2 projects already highlighted here now that you're going to hear very soon about. Before we go into that, I would love to introduce Dr. Rishi Desai, who is a assistant professor here at Harvard Medical School and member of the Division of Pharmacoepidemiology, and who's the executive director of the Sentinel Innovation Center. Rishi?
Thank you, Sebastian, for setting the stage so nicely, and welcome everybody. We are really excited to have you, really excited to share the work that we have been pursuing over the last couple of years. And over the next 3 hours or so, you will be hearing about various initiatives and salient challenges that we have faced and the approaches that we are trying to take to address those challenges. So welcome again, and I hope you will learn something new from the talks that we have for you today.
Thank you, Rishi. And before I introduce Bob, please make use of the chat function, any questions that you have, and we hope that some of the panelists will be able to respond either in the chat directly or then pick it up as a verbal comment as part of the presentation. So with that, I would love to introduce Dr. Robert Ball, the Deputy Director of the Office of Surveillance Epidemiology at the US FDA. And Bob has been with the Sentinel initiative from day one and is spearheading Sentinel from the FDA's perspective. Bob?
Thank you, Sebastian. And it's my pleasure to welcome everyone to Sentinel Innovation Day on behalf of FDA. In 2019, FDA outlined a 5-year strategy for Sentinel, and key to that strategy was the incorporation of new EHR and other types of data and advanced analytic strategies to improve Sentinel's ability to make inferences about the safety of drugs and biologics, as well as to further our understanding of real-world and evidence generation more broadly. And we're now about halfway into that 5-year time frame. As Sebastian has indicated, the Innovation Center is beginning to turn the vision that was outlined in that strategic plan into reality. And so today I'm very excited to be able to introduce the work that's being done on several of those fronts. And you'll hear today about the better use of EHR data and AI technologies for computable phenotypes, as well as efforts to improve our ability to do causal inference using real-world data in Sentinel. And while from FDA's perspective, this is very exciting for Sentinel, because it's laying the foundation for the future of Sentinel's use of better data and technologies. It's also providing deep insights that I think will be useful to the entire community that's interested in real-world evidence generation. And of course, none of this would be possible without all our collaborators. And I especially want to thank Sebastian, Rishi, all the leads of the centers, and all today's speakers. So without further delay, I'll hand the platform over to Leslie Curtis from Duke University, who will be moderating the first session. And thank you, and I hope you enjoy today's presentations.
Thank you, Dr. Ball. And it's my pleasure to introduce our first presenter today, Dr. Keith Marcelo. Dr. Marcelo is an associate professor in the Department of Population Health Sciences here at Duke and a member of the Duke Clinical Research Institute. His body of research really focuses on the infrastructure to support the use of electronic health records and other real-world data. Dr. Marcelo is a computer scientist by training, and so his early work on data mining and modeling and classification of biomedical data certainly served him well in his research pursuits. Aside from his leadership role in the Sentinel Innovation Center, Dr. Marcelo is also involved with a number of national real-world data initiatives, including the RADx UP Data Coordinating Center, the NIH Pragmatic Trials Collaboratory, and the Coordinating Center for PCORNet, where he plays a major role. He's the co-PI for the Common Data Model Core that's responsible for the support and maintenance of the PCORNet CDM. So with that, let me turn it over to Dr. Marcelo, whom I will probably refer to as Keith from now on. Keith, take it away.
Great. Thanks, Leslie. So I want to talk about a couple of projects that we are working on, really that are focused on, sort of tackling some of the challenges and opportunities around integrating electronic health record data into Sentinel. So if we go forward, I think 2 slides. And then, yeah, great. So really, as we're working to establish, as Sebastian and Bob mentioned, this infrastructure of claims linked with electronic health record data, there's a fair amount of infrastructure work that's really needed to bring these EHR data into the Sentinel framework. And so the projects that I'm going to talk about today are really come at different aspects of this. And so really the first one is focused on representing sort of information derived from unstructured data into a common data model framework. And then the other is really focused more on some of the structured data that exists within EHR and talking about identifying and sort of mitigating, mapping issues that can come with bringing in structured EHR data and sort of how to identify and what to do. So if we go to the next slide, and then one more. So when we talk about incorporating unstructured data or information derived from unstructured data into a common data model, the goal is of this project is to provide some guidance to the Sentinel network on how best to do that. And the way that we've structured this project is really in terms of three major objectives. And so the first one is really asking questions around sort of what information is important. And so, when we talk about deriving things from unstructured data, there's a whole host of concepts or just sort of things that we could try to identify or pull out of unstructured data. And so the first objective is really, what's important? What are the priority elements that, if we're going to start asking Sentinel partners to run natural language processing on all of their notes, what are the types of things that they should try to extract from that text? The second objective is starting to get a little bit of a landscape analysis around sort of the current state of the art, if you will, around natural language processing. So, what natural language processing tools are in use today across the Sentinel ecosystem and kind of how are they used? And then sort of switching gears a little bit is around, you've got the tools, but then the other question is sort of given the text that we might have available, what is it that we could, try to reasonably extract from a note with sort of, any sense of reliability. And so that's going to depend somewhat on the tool, but then also just based on sort of documentation practices across the health system. It's one thing if the tool can extract it, but is it something that's sort of documented with any reliability? So that's really the sort of the second objective of this project. And then the third is just getting down more to brass tacks and just how do we represent information derived from unstructured text? So if we're going to start to put all this in a common data model, start to use it in routine analyses, how do we go about structuring this? And so this project was originally proposed as a 12-month sprint. I think we were a little bit over ambitious in terms of that timeline. So we're going to likely extend it towards the end of this calendar year, but still trying to wrap this up in around 18 months in total. So if we go to the next slide. So this is just a little bit of the project team. So I won't get into this too detailed, but Judy Morrow from Harvard Pilgrim is sort of our liaison to the Sentinel Operations Center, being led out of Duke. Leslie is one of the co-investigators, and then have some great collaborators from Vanderbilt and Brigham and Women's helping us through this project, and then a number of colleagues from FDA are also participating. So it's a really good group, and I'm really happy for all the contribution from our co-investigators. Next slide. So when we talk about, sort of what's important, the general process that we followed for this was really to identify a list of concepts that are available, that can be extracted from commonly used natural language processing pipelines. And we looked at a variety of tools, both commercial and open source. When we were going through this, we weren't necessarily focused on like specific items. We were focused more on broad categories. So unless they were kind of called out in the documentation. So as an example, we want a tool that can extract medications in general, not one that's just geared specifically towards aspirin, for instance. We looked at the basic functionality that was provided by each tool. We didn't really dig into sort of every single research extension that's out there because there's sort of hundreds. And then we kind of stopped when we reached saturation, and we got a good enough list. And so just to kind of illustrate what I'm talking about a little bit, this is images on the right are from the sort of the Microsoft tool that exists for processing notes, and it's just sort of some examples where you would run the note through the pipeline, and it would sort of tag the different things as the concepts. And so the sort of words underneath the line there, gender diagnosis, medication name, time, for instance, those are all examples of kind of concepts that could be extracted from a note based on the functionality of the tool. And so when we had that list of concepts, we gave that to the FDA and just asked them to, one, identify anything that might be missing, because it's quite possible that sort of for Sentinel purposes, for safety purposes, might be interested in things that aren't commonly extracted from notes, so a pipeline might not exist. So looking for anything that was missing, and then assigned a priority ranking as sort of high, medium, and low. And really, when we think about the priority rankings, sort of highest priority was based on sort of two factors. One is sort of importance for drug safety studies, which is somewhat obvious. But then the other one that is really sort of is it providing sort of information on concepts that are not easily obtained from claims, because we are talking about sort of linked EHR and claims data. So if a concept is sort of readily available, it might not be sort of as important because there's sort of already ways of getting at that. And so the end product of this particular objective was a set of priority elements to be derived from unstructured text. And so if we go to the next slide. So this is just an example of some of the priority rankings. And so the way that this is organized is that the top table is sort of concepts from existing tools, and then the bottom table is sort of some of the missing concepts. And again, it's quite possible that there's a tool out there that can extract some of the things that are missing, but sort of in general, these were sort of new concepts. And so things like cancer information, because there's a number of studies that are sort of insufficient information within claims because there's not information on sort of cancer staging and things. Diagnoses, depending on the level of granularity, may be missing. Signs and symptoms, usually not in claims. And then sort of the type of diagnosis, sort of a medical history, family history, useful in certain types of studies, generally not part of claims. We start talking about sort of things that are missing. The timing and the duration of medication, that's fairly obvious why that's important for a drug safety study. Some physical findings, the indication why the drug was used, and then sort of oxygen support, which has become very crucial for things related to COVID-19 over the last couple of years. And then the last one that you can see there is sort of death date and cause. And so this one is actually marked as low. And the reason for that is mainly because some of the Sentinel data partners within their claims actually have fairly robust death data. And so it's sort of less of a priority because there's sort of other ways of getting at that information. So that's death is hugely important for studies, but just there's sort of other ways of getting at that information. So that's why it's marked the way that it is. Go to the next slide. So the second objective, what NLP tools are in use and sort of what information is in a note. So we distributed a survey to partners within the ecosystem. to understand sort of their capabilities and tools that are used. And then the other aspect of this is a chart annotation that's currently ongoing. I'll talk about it in a little more detail at Vanderbilt and Brigham and Women's, and sort of summarizing all that is kind of the end product of this objective. So the next slide. So the sort of initial results of the capability survey. So we distribute this survey to 18 data partners within Sentinel, and then sort of 8 partners that are affiliated with the Innovation Center. And then of those 22, we got a total of 17 responses. And so, what we found was that 12 of the partners were using natural language processing in some capacity. It was roughly split between sort of 50% project-specific research and then sort of 50% for research or just somewhat vaguely defined operational purposes. And we left it to the partners to kind of define what that meant. But really, it's sort of like our using it in routine practice as opposed to project-specific. That's what we were getting at. There was a wide variety of tools that were in use. I don't know that there was a whole lot of there really wasn't a lot of commonality. I think everybody was using something slightly different. And then there was also a fairly high variety in terms of the types of notes that are processed, whether it was sort of, they were doing all progress notes, pathology notes, radiology notes, discharge summaries, things like that. So again, wide variety in terms of what people are doing currently. And then the scope of the concepts that are being extracted was also fairly varied. And so the sort of highest percentage was sort of 3/4 being able to extract diagnoses. That was the highest. And then really sort of, it kind of went down from there. A handful of other concepts extracted by, greater than 50% of respondents, and things around sort of cancer site and histology, smoking status, and then kind of signs and symptoms. And so, this is actually, it's informative if we think about how do we start incorporating natural language processing in Sentinel, because we have to understand, again, the current capabilities of the different partners. And then again, ask everybody to start extracting signs and symptoms, for instance, either providing tools or guidance on how to go about doing that. So it's sort of good information around the state of the state. So go to the next slide. So with the chart annotation, really the motivation behind this is it's a future state where Sentinel partners have access to EHR data, and they've processed. Really we're saying we're moving to a state where they've processed sort of all or some of their clinical notes through a pipeline. It's not saying that there's going to be projects that need to develop new pipelines or new classifiers, but we're really thinking like, what is it to rely on sort of a stock NLP output? Like, what kind of analysis could you do with that? And so part of that is just understanding what could be extracted from the notes. So the issues to consider, sort of what notes have been processed, what's the time frame that's been covered. And so as an example, if we're looking for a patient with a history of a myocardial infarction, if a patient had an MI 10 years ago, do we have to go back and process 10 years worth of notes? Is it something that we can assume it's mentioned at every visit, new visits with visits with a new provider, sort of every 2 years after event, sort of just like at what point, are we seeing that information, and then, does it continue or does it start to drop off? And so that kind of thing is what we're starting to get at with this chart annotation. We're not going to fully solve that, sort of answer that question, but that's sort of the motivation behind it. So if we go to the next slide. So we're focusing on 2 use cases, hospitalized patients with COVID-19 and cancer. And again, it's just to understand the information in the notes, not really to develop a pipeline or a classifier. So go to the next slide. So with hospitalized patients with COVID-19, basically we're looking at patients with an inpatient, with a hospitalization in inpatient visit with an admitting diagnosis of COVID-19. And so the specific codes behind that between April 1, 2020 and then the end of last year, limiting to patients who are greater than age 18. And sort of 2 cohorts that we're choosing. One without a billing code for supplemental oxygen, and then another cohort with a billing code, just selecting sort of 50%, 35 patients, 50-50 for each one. And then the primary analysis is pulling the discharge summary associated with the hospitalization, and then annotating the sort of high and medium priority concepts sort of identified by FDA. So things like oxygen use, conditions, medication exposure, and related metadata for sort of timing, dose, things like that, smoking status. And then proposing to do a secondary analysis on a subset of patients, just running a query on sort of all their other notes, looking for keywords related to oxygen use, and then reviewing the note around that keyword, and then just sort of describing the oxygen use. And sort of some of the rationale for this, because you might be asking sort of why are we going about it this way? So, you know, we realize that discharge summaries are not going to have sort of the full detail related to oxygen use. If we really want to know sort of everything related to oxygen, we would actually be focused on sort of respiratory therapy notes. So what we're doing is we're looking at the discharge summary and then just basically trying to characterize the degree of missingness that we see by only looking at the discharge summary. And the reason that we're looking at the discharge summary is, you know, it is pretty rich. It doesn't have everything, but it's pretty rich. And if we're going back to the use case of let's do an analysis on sort of pre-computed information extracted via natural language processing, it's more likely that sites are going to have processed their discharge summaries than some of the specialty notes, because you sort of get a bigger bang for the buck. You know, if you're talking about sort of processing hundreds of millions of notes, you're going to sort of start with those that are give you the richest information. So the assumption is that, you know, more would have discharge summaries than not. And then, you know, stratifying by sort of billing codes for supplemental oxygen just helps us ensure that there's a mix of patients who did and did not receive oxygen compared with just sort of a completely random sample based on, you know, just who was hospitalized with COVID. So that's some of the rationale behind that. Next slide. So the next use case is really looking at something related to cancer. And the way that we tried to do this was essentially looking at patients who were newly diagnosed, newly prescribed, in this case, an order for Darzalex, and just sort of selecting 30 patients from that cohort, and then annotating the physician note associated with the visit where the patient was prescribed the medication. And then sort of annotating some of the selected concepts. And really the motivation behind this is if we were to look at sort of the information contained in the note where the patient was newly prescribed the drug, would we find enough information to determine sort of the reason, sort of the indication behind the drug? And so this is the label is here on the right, and sort of the different reasons why, you know, somebody might be prescribed the drug. And just, you know, if you were to develop a classifier that wanted to sort of bucket somebody into one of those 4 different categories, you know, what kind of information is in the note, and would it allow you to sort of get at that? We're not actually developing the classifier as part of this project. That's out of our scope, but really just starting to understand kind of what information is in the note and what do you get out of it. So that's sort of the rationale behind that particular use case. We go to the next slide. And then the third objective is this is still ongoing, is just how do we best represent information from unstructured text? So we're assessing current approaches from other common data models, NLP tools, and so forth, describing some of the trade-offs in terms of the ease of querying, the burden on the partners to just sort of populate information. And then we talk about sort of representing information. What are the different strengths and weaknesses of different terminologies? Sort of the end product of all that is a set of recommendations for the operations center as they think about, you know, how to turn this into a sort of just general infrastructure. We go to the next slide, sort of switching gears. So now we're going to go from sort of unstructured data back to, you know, sort of structured data within the EHR, and talking about identifying and mitigating mapping issues. So if we go to the next slide. So the goal of this project is again, to just assess the mapping, develop some quality metrics, and I'm going to use sort of metrics, but it's kind of interchangeably with queries. So when I talk about metrics and queries, they're really kind of the same thing. So the objectives here is just developing procedures to assess the mapping between the source EHR data and reference terminologies, looking at 2 specific or really 3 specific domains: laboratory results, medication orders, and then administration, and just sort of characterizing the severity of issues. And part of the reason that we're looking at those specific domains is really that, you know, within the EHR, data are often not captured in sort of source terminologies, and labs and meds are very important. Things like diagnoses and procedures tend to be a little more robust in terms of their mappings, just because those are used for billing purposes. So there's a bit more scrutiny there. Labs and meds, not so much. And so I'm focusing on those. And then the other thing that we're doing as part of this is really sort of standardized queries that are this is a very sort of long-winded way of saying looking at provenance. So if we start to look at the provenance of the information, so like clinician entered diagnoses versus billing diagnoses, or medication orders versus administrations, and starting to use that to try and identify basically identify quality issues that might appear. And so this work is sort of supplementing an existing Sentinel project, the data quality measures project, by just sort of new strategies for assessments. We go to the next slide. Work is being led out out of Duke. Judy is again our Sentinel liaison, and then sort of partnering with two sites within the PCORnet network, the Mayo Clinic and University of Florida, to help test out some of these queries. And then the other thing I would mention is that Duke is also sort of a data contributing member to this project. Next slide. I touched on a little bit of this previously. So the motivation is harmonizing data sources. So again, starting from the EHR sort of custom source, and then harmonizing to a standard, and just trying to understand some of the issues that can appear. And I will say that the examples that I'm going to talk about, this comes from from PCORnet, but these are really not specific to PCORnet. They sort of they exist kind of regardless of where you're getting it from, because they're inherent to EHR data. So if we go to the next slide. So this is an example of sort of how medication might be represented. And so this is within PCORnet, medication orders and most administrations are coded using a standard called RxNorm, which is it's an interoperability standard maintained by the National Library of Medicine for just representing medication information at various levels of granularity. And so what you can see here on sort of the top right corner is a string for Augmentin XR 12-hour 1,000 milligram tablet. And then sort of everything, all the rows sort of down the table are just sort of valid RxNorm representations for that same medication. And the take home here is really just that those representations could go from sort of very, very detailed and specific down to very, very generic, to the point that they're basically unusable, right? So you can have a semantic branded drug level that represents it. It describes sort of the ingredient, the strength, the dose form, the brand name. You could also just sort of have the dose form, so it's extended release tablet. Those are sort of valid RxNorm codes for the same medication. And the reason that we pay attention to this is, again, because sort of EHR data may not be coded to any standard in the beginning. It may just be sort of an internal kind of internal reference. And so it's likely that the information between sort of source and Sentinel, it could potentially, sort of hop through one or more reference terminologies. So that's sort of why we're thinking about this. If we go to the next slide, I'll just say in PCORnet, we've worked very hard to try to get sites to sort of map to a preferred tier for different terms. So the ones in green at the top are sort of our preferred ones, because they basically encode the most information. But there may be cases where you sort of have to go to a lower tier and sort of different strategies for focusing on that. Although that's not the specific purpose of this analysis. Next slide. So to just sort of give an illustration of sort of some of the quality issues that can occur when you're going from your source to sort of a reference standard. So these are sort of real results from a site within PCORnet. And the way to look at this is we took sort of the top medication, basically we took the medication data within their prescribing table in this case, and we looked at sort of the highest volume number of records based on just kind of the string that describes the medication, the raw string from the source system. And those are the sort of the three columns on the right side. And then we looked at sort of information that had been, basically the records as they've been mapped to an RxNorm code. And we took the highest volume based on sort of the RxNorm value, and just sort of lined them up together. With the thinking that sort of the highest volume by name and the highest volume by code, there should be sort of some correlation there. And when you look at the comparison, you can see that in general that's the case, but not always. There's some issues that appear. So sort of the highest ranking is sort of no code and sort of no string, so that you can just kind of ignore that top row. The second row is sort of, you know, a string of sodium chloride within the EHR, and then sort of there's a sort of a more specific code that was set for RX norm. So that's pretty good. You can see the percent agreement in terms of the number of records. It's about 80%, which sort of tells you that there's probably more RX norm codes for sodium chloride sort of elsewhere within the system. If you sort of step down, what you can see is the sort of raw string is acetaminophen 300 milligrams, and then with codeine, right? So a combination of Tylenol with codeine. But when you look at sort of the corresponding mapped code, it's actually just acetaminophen, and you can sort of see the other highlighted examples in this table in sort of the yellow color are similar sort of scenarios where you've got a multi-ingredient on, you know, within the EHR, sort of mapped to a single ingredient within RX norm, and so that, you know, that's an issue if you're trying to use these information for studies. And so those are the kinds of things that we're trying to uncover with this particular project. If we go to the next slide. So the first objective here is just methods of assessing structured data. So queries to assess the mapping of orders for medication orders, administration's lab, limiting analysis to the top 200 by volume, just for sort of ease of analysis. And then basically for each medication and lab, we're just sort of generating statistics on sort of all the different combination of the different fields. And so this is just a subset of what is sort of we'd expect to come back. And so this is a medication, sort of the raw name, you can see there on the left column, and then the different RX norm codes that it's mapped to. Again, mapping to more than one is not necessarily a problem. In some cases it might be, but in others it's not. For this particular one, it's not. The first set of codes are for the brand name, and the second set of codes are for the generic, or it might actually be vice versa, but it's sort of describing the same thing. And then just sort of looking at sort of the like dose form and the dose and things like that, and just trying to uncover issues. With this particular one, I think this is maybe less of a mapping issue, but this actually sort of illustrates a potential issue in sort of a CDM sort of representation issue. You can see the dose units that are sort of in the source system are all mapped to other, meaning there's not a clean sort of mapping, right? You may have milligram within your common data model to describe the dose unit, but then you've got sort of more specific in your source data. So the question becomes, you know, how do you want to handle that? Do you expand the data model? Do you provide some additional guidance and things like that? So that's some examples of that. If we go to the next slide. And then just evaluation, sort of different statistics, and then basically using all that information to describe sort of the severity of the issue. And then, you know, sort of most severe is when you've got sort of either the wrong code, completely wrong code, like a lab test, wrong one is chosen, or, you know, sort of multi-ingredient to single ingredient, or vice versa, and then sort of stepping down in the severity. So, you know, working to basically run the analysis on all of that. So if we go to the next slide. So sort of the next objective for the project is really looking at kind of differences on provenance. And so a couple of examples here, again, from PCORNet. This is really looking at 3 different medications and just sort of looking at the number of orders versus the number of administrations within the system. And really the example here is to just, and each of these dots represents a different data mart within PCORNet. And again, not necessarily saying anything about necessarily on what we would expect, but just looking at sort of the differences across the partners to see, you know, are we starting to see outliers? And so for adalimumab, you know, you can see in the top, it's basically, you know, there are sort of no administrations compared to orders, right? So basically it's orders, there's sort of no administrations within your data model. If you look for donepezil, it sort of tracks on a kind of 1-to-1. It's not exactly, but that's the sort of trend. And then you can see in the very top, right, there's one where you have sort of 3 times as many administrations as you do orders, which could indicate a quality issue. And then same for sort of ketamine, where you've got sort of a couple of different issues that might be there. Some where you've got sort of administrations with no orders, or you've got sort of orders but no administrations. And again, you could start to use that information to help tease out some potential sort of quality issues in terms of sort of loss of data feed and things of that nature. So we go to the next slide. So again, the sort of the objective is just sort of queries to start getting at this. And so queries support comparisons of records based on provenance, so orders versus administrations, build diagnoses versus clinician entered to identify data issues. And so the way we're going about this is defining specific conditions and sort of associated concepts like diagnoses, procedures, meds that you'd expect to see related to that condition, and looking at values within the cohort and the population as a whole. And sort of, again, the thinking that looking at condition-specific information might sort of elicit information that's sort of masked by population trends. And so this query is being distributed to a couple of data partners, and then we'll use those for results. And so just as a couple of examples, I think some of the font gremlins got with these tables, but the top one is an example of the type of information we might retrieve looking for provenance. So looking at a COPD cohort with a CAD diagnosis, and then starting to look at provenance, sort of ordered diagnoses versus builds versus, if somebody's running NLP, like we sort of talked about in the previous project, looking at number of projects, number of patients, number of records sort of by year, and then also looking at sort of the entire cohort. And again, you start to see sort of variations over time and things like that. And then similarly at sort of medication information, so prescribing versus administrations versus sort of patients with records in kind of both tables, and you start to see changes over time. And again, just starting to see what we uncover, and then how we can inform sort of guidance for Sentinel as they think about incorporating EHR data, and then partners as they think about sort of cleaning up their data model. I think that's my last slide. And so a few minutes I think we have for questions.
Great. Thank you, Keith. That was excellent. And certainly if you have questions, please post them in the chat. I'm happy to take those to Keith. Keith, I was really, and we have been struck, I think, by the amount of heterogeneity that exists. And I'm talking specifically about the first project, right? The amount of heterogeneity that exists with respect to the use of NLP, the tools that are used for NLP, and what they're used to do within sites. And that's surprising, because often when we talk about NLP, we talk about it as though it is a sort of a thing that is used in the same way across multiple institutions for the same things. Can you talk a little bit about, maybe just reflect on that? And were you surprised by the level of heterogeneity that we found? And then I have a follow-up question, but maybe start with that.
I was surprised, but then I guess also not surprised in a sense, because I think, so part of this is, the tools have been maturing, and they've really come a long way over the last, few years. And I think, traditionally NLP has been used where it's sort of like a faculty or, an investigator sort of interested in NLP, and they'll sort of use it for sort of purpose-driven activities. And that was certainly reflected in a lot of what we've seen. And what I would say over the last few years now, it started to become more like a service. And I think some of this may just be that it's sort of adoption is lagging somewhat, and you can always blame the pandemic for sort of putting the brakes on sort of these major expansions. But we are now seeing that some of these tools are like you can on the Amazon cloud or the Microsoft cloud, it's a tool that you can just sort of apply to all your notes. You can run it. It's like 3 cents a note to process or whatever it is. So it's now become a little bit more commodity. I think because of that, we are going to have to pay attention to sort of how it's used in the performance and all that kind of thing, because it's not, it doesn't perform equally well on sort of all kinds of notes and things like that. So what I would say is in years past, we may have been talking more around sort of, you know, how do we want the tool to work and sort of what's the pluses and minuses there. But now we've got it where they're just sort of available. People are going to start to use it for various purposes, and we kind of have to understand sort of what some of those differences are. So so that's a very sort of long-winded answer to your question, but I think that was sort of some of my takeaway from those.
Yeah, it's a great answer. And you managed to actually anticipate what my follow-up was, which was, and how has it changed over time? What do you see? I guess maybe just one last question before I turn to the next project. Are there specific, as you look at how tools are now much more widely available, what do you see as sort of the critical issues that we have to be able to address in order to leverage them for these kinds of multi-institutional efforts?
Yeah, I think that there is sort of within, certainly like Sentinel is going to face this problem, but I know other sort of NIH initiatives are grappling with this now as well, sort of around like the Recover initiative for sort of long COVID and even N3C. Starting to think about, you know, are we going to have everybody do their own sort of like, do you just sort of set a threshold and say like, if you can use your own NLP tool as long as it's sort of, you know, it's above this bar, right, for whatever sort of things we're asking you to extract. That's one approach, and that's certainly a fine approach. The other would be, you know, we're really going to try to start to ask people to sort of use the same thing, whether it's you know, there's sort of containerized services and all that kind of stuff that you can think through from a technical implementation. But we kind of everybody should process their notes in the same way. So that's sort of the other option. And I know that, again, there's pluses and minuses to both, but I think that's going to be sort of where we start to go now in terms of this is I think people are starting to say like, let's put, you know, natural language process through its paces. Let's start to try to do it at scale. And again, it just it ends up generating sort of a different set of issues than maybe we've had to grapple with in the past. But you know, it'd be great if we can start to get some alignment across these different initiatives and start to, you know, reuse some of that infrastructure. Yeah.
So I think we might have time for one quick question, and I want to turn to the second project that you described, where you know, you showed a slide with an example of quality issues with mapping of medication data, right? And I think what might confuse a lot of people is that you also described RX norm as a standard, which I think to some of us, we might think a standard means that everyone uses it the same way, right? So, and what you showed is that they don't. And I know we're probably at time, so I wonder if you just quickly want to mention that, respond to that.
Yeah, and so, again, within sort of the EHR, every sort of EHR is different, but many have their kind of internal medication databases that you can kind of purchase from different vendors, and they're going to have an internal standard. When you want to start to share information out to something else, there's the sort of NDC, there's sort of GPI, there's different sort of standards that you can use. Since you don't know what's going to be dispensed, NDC might not quite be appropriate because you don't know the exact, formula. With CoreNet, we did RX norm because it's sort of the standard promoted by the Office of the National Coordinator for Health Information Technology. But people go about it in a different way, whether sort of using mappings provided by their database vendor, they're sort of mapping directly, again, sort of NL, tools by the National Library of Medicine. And so I think part of this is understanding some of that what's the guidance that you want to provide people. But I think actually the other thing here is, I think we've all assumed that like you start with source data, somebody gives you a code, right? That's good enough to do what you're going to do. I think what we're discovering is you actually sort of need to understand, get back into that source data and look at some of those, like the raw text strings that exist in that database to validate, at least, in the beginning, sort of that, that mapping is correct. I think we've assumed that, I don't need to see that sort of raw EHR information either because it's sensitive or whatever, but it's really actually pretty important for, for quality purposes.
Great. Well, thank you. Thank you, Keith. That was a great presentation. And with that, I will turn it over to our colleague, Dr. Jennifer Nelson from Kaiser Permanente, Washington. Jen?
Thanks, Leslie. Hello, everyone. I'm Jen Nelson, and alongside Leslie, I'm one of the Innovation Center leads for Sentinel, and I'm also Director of Biostatistics at our Health Research Institute at Kaiser Washington. And I'm very pleased to introduce our next session, which is going to highlight a variety of connected Sentinel Innovation Center work on methods and applications in computable phenotyping to aid medication safety investigations. Our first speaker will be Dr. David Carroll. He's an associate investigator at Kaiser Washington, and his research uses clinical NLP methods to extract information from unstructured clinical text to support precision phenotyping. And through collaborations with researchers who bring complementary expertise in clinical informatics and statistical domains, he's applied precision phenotyping methods in the areas of cardiovascular disease, cancer and cancer risk factors, substance use disorders, mental health conditions, and medical product safety surveillance. He's also currently co-leading the Sentinel Innovation Center's Future Engineering Core. Second, we'll hear from Dr. Joshua Smith, who's an assistant investigator of biomedical informatics at Vanderbilt University Medical Center. His research also focuses on the use of NLP as well as data mining and machine learning at the intersection of data science and patient safety. His past work includes adverse event discovery and detection from unstructured clinical notes, clinical decision support for pediatric pneumonia using NLP on radiology reports, as well as mining of unsolicited patient complaints to address malpractice risk with the Vanderbilt Center for Patient and Professional Advocacy. Dr. Smith is also current co-lead of the Sentinel Innovation Center's Detection Analytics Core. And finally, we'll have Dr. Adi Bejan, who is a computer scientist who works in a research area that lies at the intersection of biomedical informatics, NLP, and applied machine learning. And in his current role as assistant professor of biomedical informatics at Vanderbilt University, Dr. Bejan develops advanced data mining technologies for analyzing big collections of medical records to identify phenotypes of interest and facilitate clinical and translational studies of large patient cohorts. So we've got a really great slate of speakers lined up, and I'll start by turning it over to David.
Thank you, Jen. It's a pleasure to be here today, and I'm going to turn on my video just a bit so you can say hello and see me, but I'm going to turn it off to conserve some bandwidth here. And we'll get started with the next, please. So here's our outline. I'm going to talk a little bit more about the motivation for computable algorithms in Sentinel and some of the limitations that are leading us to incorporate EHR data and machine learning methods. Separately, I want to talk about this concept of scalable algorithm development, where the thing that's scalable is the development process itself. And that's important for some work that it's going to be shared by one of the other speakers, Dr. Josh Smith and Dr. Adi Bejan. And then in the third section, I'm going to talk about some work we've done at Vanderbilt and at Kaiser Washington, developing filters for outcome identification, and what these are, and some new twist on this that we're proposing. Next slide, please. Many of you are familiar with ARIA, the FDA's active risk identification and analysis system for post-market safety surveillance. ARIA has two main features, as shown here in this graphic. One is a predefined parameterized set of analytic tools, and tools can be used to query data and to conduct an analysis. The second is data in a well-defined common data model, the Sentinel common data model. And these two features together allow Sentinel to quickly and easily execute distributed programs on data from over 100 million lives that are in this distributed database. ARIA allows many safety issues to be investigated rapidly and at low cost with these tools. Next slide, please. But there are some static analysis that's a few years old now, but it's still useful for framing the problem. and I won't get into the details, but it's shown here in these boxes, about 2/3 of the studies where ARIA was not sufficient for conducting a desired safety study. The issue had to do with problems in attempting to capture the outcome of interest. So we now know from work in Sentinel and other work published in the literature that there are ways to address some of these insufficiencies. Next slide, please. So accurate identification of some outcomes or covariates requires information that is only available in rich EHR data, the sort that Keith has been talking about and that you've heard about from some of the other speakers already today. A lot of this comes from clinical notes extracted by NLP, but it could, even labs data, which are now coming into the Sentinel Common Data Model, can be very valuable. We've seen as in some examples here in identification of acute pancreatitis, it's difficult to do that without labs data, but if you have labs data on lipase, you can do it very well. Another example is anaphylaxis, a much more challenging outcome to identify, but many important features, symptoms, and characteristics that help identify true anaphylaxis can be extracted from electronic health record data and from clinical notes. Now, because these EHR data are much more granular and much more high dimensional, it often is best to use machine learning models to actually identify the signal that's in those data. And of course, machine learning models are being used commonly in health and other domains as well, but we've all helped. Next slide, please. Okay. So now development, and really there's these three features that I want to emphasize. First is efficiency. So we by efficient algorithm development that we have to be able to do this at a reasonable cost and in a reasonable amount of time, where the cost drivers are things like the salaries of expert personnel to engineer features, to create gold standard and those sorts of tasks. Often that is the most time-consuming and expensive aspects of algorithm development. Secondly, scalable algorithm development has to be portable. And by that, we simply mean that if you develop an algorithm in one setting, it must be something that you can transfer to a second setting and implement it through shareable tools with minimal or no local tailoring, and in a way that anticipates the local data systems and local needs. We've heard already how there's a fair amount of heterogeneity across different settings in the EHR data. A portable tool ideally would be able to accommodate those. This turns out to be a tough thing to do well across all phenotypes. And so it's one of the things that we're going to come back to and see that there may be some approaches for addressing this portability problem. And then finally, replicability. We rely on the algorithms to perform comparably across settings, and then also within a single setting to perform comparably across time or across different cohorts. So the three of these together make up what we're calling scalable algorithm development. And if we can produce algorithms in this way, then we can both keep pace with the demand for safety analyses, and we can also produce results at a reasonable cost and that are comparable across settings. So that's all I'm going to say for now, but later on in Josh Smith's talk, you'll want to come back to this thought about scalable algorithm development and why it's important. Okay, now on to the main part of the presentation here. If we go to the next slide on filters and their role in identification. If you could advance one more click, and then a second click, and then pause there. Okay, great. So filters, if over here on the right, we see the graphic of this big gray circle that's so big it can't fit on the screen, representing all encounters and all patients. The health outcome of interest to a safety study is often a very, very small set of those outcomes in patients. And so one of the first things we want to do in creating an algorithm to develop that health outcome is identify some kind of filter that can help us identify, greatly reduce the amount of patient encounters we have to look at, but hopefully capture all or most all of the outcomes of interest. They're usually expert specified sets of diagnosis codes. It could be procedure codes, could be one code, could be a small set of codes. They presumptively identify the patients. Now, it's presumptively is an important term here because the filters can also include patients who don't have the outcome of interest. And then within that presumptive set, the goal of the algorithm itself is to accurately separate those who are true cases from non-cases. So some useful characteristics of filters, they should have strong face validity. They should be relatively simple, which will help them be generalizable, and they need to be generalizable across settings. Two other important characteristics is they should be highly sensitive. So they should capture most of the true cases of that health outcome. And this would help with things like avoiding selection bias. And then they should be reasonably specific so that they don't require boiling of the ocean. We can focus our work on a modest set of patients and then just distinguish the true cases from the non-cases. If we could click one more time, we'll see here in this green circle an example of what a reasonable filter might be. You can see it gets the vast majority of the true health outcomes of interest. It also picks up some that don't have the outcome of interest, but those are the ones where the algorithm is going to decide of those that are inside the green circle, which ones are blue and which ones are gray. Now, outside of the green circle, you can see there's a crescent shape there of patients who have been overlooked. A goal of high sensitivity filtering is to try to figure out if we can somehow augment our filters to bring in some of those overlooked patients. But it's not as easy as it might think. If we could click once, you could define a filter that would be very inclusive and it would get everyone or most everyone with the outcome, but it would also add so many patients that the burden of processing the data would be not worth the trouble. If you click once more, we'll see another approach would be to choose a filter that was highly, highly specific, but not so sensitive, in which case we wouldn't have the problem of false positives here, but we have a huge false negative problem. And so click one more time, please. So what we're going to do is show you based on, oh yeah, so the two solutions in black are not very good filters. One more click and we'll introduce here that the work of two teams, one at Vanderbilt and one at Kaiser Permanente, led by Josh Shelton and Smith, have proposed this high sensitivity filtering approach. And I'll just give you here on this slide a brief introduction to it, and then we'll see how it works in practice in the case of COVID-19. So the objective, as I've already said, is to be fairly sensitive and with reasonable specificity. And that's the objective of the traditional filter. The high sensitivity filter then is to see if we can recapture some of those lost patients or encounters that the traditional filter overlooks. But even in doing that, again, we have to do it with a modest increase in overall sample size. And it shouldn't require so much effort that it's more trouble than it's worth. So we have to find a way to do this with a reusable tool. Now here, down in the bottom section here, is a brief summary of how this method works. If you want the details, you can look in the an abstract that we have. It's still under review. Hopefully this will be presented and published soon. The first thing to do is, among those patients who you've filtered, so if you think back to our green circle, everyone that was in that green circle, you divide them up into two groups. One is people who ever met the traditional filter, and the other is who never qualified by the traditional filter. So, I'm sorry, this is actually is something we do to all the people in the entire data set that's available to you. So you can have these two groups: those who meet the traditional filter, those are inside the green circle, and those who never would meet the traditional filter within the period. Then, with those two groups, you just calculate a bunch of ratios for all possible codes of interest, diagnoses, procedures, medications, labs, whatever. And you look at those codes that are at least 10 times more common in the ever group than the never group. And you also look at how many additional patients each one would bring. And you do, with that kind of filtering, you can produce a list that's modest in size, validity, to see, oh yeah, looks like some of these codes might be useful.
David, I just want you to be aware you're cutting out, and I don't know if there's something you can do to improve your connection.
And oh, I am so sorry. I am going to dial in on a telephone. Okay. Okay. I'm sorry. It's going to take about 30 seconds.
Thanks.
Hey, I'm back. Can you hear me?
Yes, we can hear you. Thank you, David.
Okay, my apologies for the Wi-Fi issues here. Jen, how much of the explanation did I get through that you could actually hear?
We got through most of it, so I don't think we missed too much.
Okay.
So you can pick up where you left off, I think.
Okay. So good enough. So let's go on to the next slide, and we'll see in a real example how this might work. So here's our health outcome of interest. It is COVID-19 disease, symptomatic COVID-19 disease. And we have up here in the upper left a traditional filter that could be specified by experts. I mean, this one came from a published article. It's actually a little dated because today a traditional filter would include about 5 or 6 different diagnosis codes. But at the time, this was a reasonable set, and it illustrates the point well. If you click once, we'll see the first code for COVID that was available before April 1st, 2020 might get a good many patients, but not all. Click again, we'll see the U07.1 diagnosis code is the one that's mostly being used. It gets a much larger set. And then click again, the third code is for history of COVID-19. Maybe this is super specific to COVID-19 patients. You only get it if you really have the disease. It's not very sensitive. Nevertheless, it adds a little bit that the other codes didn't add. So this is what we would get with the traditional filter. If we could click once, we'll see another view of this where you have in the middle, the light blue would be the true cases that you want to find. The red slashed ones would be the false positives that the algorithm would need to separate. Let's click once more. And then on the right there, on the darker shaded, are the overlooked cases, the ones that are missed by the traditional filter. If we could click again, just to remind ourselves here, the job of the algorithm would be to separate the red from the light blue. But one thing we're asking is whether or not we might be able to improve our sensitivity by recovering some of those lost cases or patients in the crescent. Let me click again, please. So the approach, as I mentioned, was to see by looking at diagnoses, procedures, medications, labs, anything, any structured data and that's easily accessed. And if you look at in those two groups, anyone who was ever qualified as by your traditional filter versus patients who never were qualified, and then you looked at those that were those ratios were at least tenfold for the ever qualified. Click again, please. Now you might see that you could pick up various other diagnoses, maybe some procedure codes, some medication codes that are very specific to treatment of COVID or your health outcome of interest, some labs that are specific to the health outcome of interest. And, you know, in this generic sense, then click one more time, please, you would see that now we've expanded the light blue section a bit. We've also expanded the red section, the false positives. And so the question becomes, you know, if we can recapture some of the overlooked patients, how many were we able to capture? Was it worth the trouble? And at what cost in terms of additional processing or data burden? So now we can go look at this in the context of COVID-19 or the actual data that we looked at. If you advance the slide, please. Okay, and this is our last slide, but I'll introduce you to it here. In the two columns are the two data sites, Vanderbilt's on the left in the gold and Kaiser Permanente, Washington on the right in the blue. All of the pie graphs in here are actually to scale. And so we can see there were many more patients coming from Vanderbilt than Kaiser. The dark shows true cases and the light colors show cases that were in the filter, but they turned out to be false positives. And then importantly here, the top row is what we would get from the traditional filter. So we can see here on the left, there's almost 21,000 patients from Vanderbilt. 90% of them turned out to be true cases by the traditional filter. Almost 7,000 patients from Kaiser Washington, about 71% were true cases. And that would be the data set that we would train our algorithm on to separate the true cases from the non-cases. What we get by going through that high sensitivity filter approach I described is what we see on the bottom. We added about 4,500 patients at Vanderbilt. It turns out based on estimated fractions based on real chart reviews, just over half of those would turn out to be true cases. So it looks pretty encouraging there. And on the right, we see we added about almost 1,500 patients at Kaiser Washington. About 38% of them turned out to be true cases based on estimates from chart review. And so this then summarizes in a single picture what this high sensitivity filter approach is, how it was applied in this one case, and you know, we'd certainly be interested in any questions or feedback folks have and how this may or may not fit well in other outcomes of interest. That concludes my presentation. I want to thank you all for your attention, and back to you, Jen, for question and answer.
Thanks, David. I think we'll move on to our second speaker and save questions till the end of all three speakers. So with that, I'll turn it over to Josh.
Hello, everyone. So today, next slide, I think. Nope, keep going. There we go. Back one. So I'm going to talk today about the same thing David's talking about, sort of. It's a continuation of the work where he left off. And this is all part of our project, Advancing Scalable Natural Language Processing Approaches for Unstructured EHR Data. And again, the work was performed at Vanderbilt University Medical Center and Kaiser Permanente, Washington. You can go on to the next slide. So our goal is to improve cohort identification for Sentinel by utilizing EHR data with a focus on COVID-19. And as we all probably know, COVID-19 severity can range from asymptomatic to deadly, and in between there's a wide variety of symptoms. Identifying complex phenotypes like COVID-19 can be a labor-intensive process. Traditional computable phenotype algorithms have typically been developed using time-intensive expert curation and manually annotated gold standard training sets with high cost and limited scalability. Unlike traditional methods, phenorm is an automated approach for creating computable phenotypes using natural language processing and machine learning, and low-cost silver standard training labels, which I'll go into in a little bit. Phenorm has been demonstrated to perform well for various chronic health conditions. And in our study, we wanted to apply Phenorm to COVID-19, an acute condition, to investigate its feasibility for rapid phenotyping, not just here, but for use in other post-market safety studies. Next slide. You can click one more time. There we go. So why are we looking at automating phenotyping methods? Primary reasons are, as David said, scalability and replicability. And as he mentioned, scalability is a combination of efficiency and portability. Is the method reasonable to implement in terms of time and cost? And can it easily be implemented in diverse real-world settings? Unfortunately, manual approaches can be burdensome and slow and often require substantial expertise. This includes manually curating structured covariates. Click, please. Manual implementation process, including assembling corpuses, creating an expert curated gold standard, and manually engineering NLP covariates. Next, we have manual chart review by experts, I think one more, and manually curating NLP dictionaries. So that's identifying words or terms, concepts that are important for the phenotype in question. And all that is normally very manual, multi-step process involving a number of experts. Next. So replicability is the other goal for applying automated phenotyping. That's to reduce operator dependence whenever we implement it at a new site, making it easier to implement the algorithm developed at one site at other sites. Next. One approach, or our approach here, is the use of hybrid solutions, such as Phenorm that I mentioned, that can automate some of this process. Next slide. You can click again one more time. So there are a continuum of development approaches that we could use. In the manual process, like I said, development's expert driven with a heavy reliance on manual engineering and gold standard labels. An automated process, they're data driven and rely on silver standard labels, which require little or no manual engineering. Manual processes are also often slower and have substantial operator dependence, whereas automated processes can be faster and have little to no operator dependence. Next. Some of these methods published in the last few years include AFEP, automated feature engineering for phenotyping, SAFE, surrogate assisted feature extraction, Phenorm, phenotype algorithm normalization, and PHECAP, which is a phenotyping comment approach. Next slide. We selected a Phenorm to use in this project. It's described in this paper in the Journal of the American Medical Informatics Association by Xing Yu and others. It's from 2018. It's called Enabling Phenotyping Big Data with Phenorm. I direct you here for a discussion of the details, but we'll go over the basics on the next slide. So how does Phenorm work? Click once, I think. So we start with our EHR data and use a filter to identify patients of interest, including cases and not cases. That's that high sensitivity filter approach that David just talked about. We filtered on cases that had ICD codes for COVID-19, as well as other codes, procedures, labs, problem lists, problems. That's the high sensitivity filter approach, and that gets us our initial cohort. Next. The next steps involve extracting structured and unstructured data from the EHR, including defining an index date for the condition and a catchment period, which is how many days and weeks worth of notes we want to include. We also defined our silver labels here, the silver labels being surrogates or best guesses for what structured data can be used to identify the condition, in this case, COVID-19. Next. Next, we needed to use what we're calling an NLP dictionary of concepts or terms that are important or relevant to the condition in question. We developed this dictionary automatically, or mostly automatically, using AFEP, the automated feature engineering, which I'll describe in the next slide. Next. So after the NLP is complete, that we identify a list of concepts that show up in these notes. And then we transform that data into feature vectors or counts of the occurrence of the concepts that are important. In this case, important meaning they were in our NLP dictionary. Something to note here, this figure also describes phi-cap, which is slightly different than phi-norm, and that it uses some gold standard data as well as structured data to make up patient feature vectors. Since we're using phi-norm, our vectors are made up entirely of NLP data. So are these concepts present or not, and how many times did they show up in these notes? Next. So finally, the algorithm generates a predicted probability of the patient being a case or having the phenotype or not. Next. And while phi-norm does not require gold standard data to create this phenotype algorithm, we did want to evaluate its performance. So we did do some manual evaluation on some cases and controls, which I'll show you in just a second. Next slide. First, though, a brief description of that NLP dictionary created using AFEP that I mentioned. So instead of asking experts to use their knowledge and chart review to determine the important terms, AFEP mines important terms from knowledge base articles on a topic of interest. So the assumption is that NLP extracted information that's needed to accurately distinguish between phenotype true cases and non-cases can be obtained via this dictionary of commonly occurring NLP extracted concepts that appear in multiple knowledge base articles. Here we see, say, 5 articles of interest for a given topic or phenotype from sources such as the Mayo Clinic, Medline Plus, Medscape, and others. Next, one more time. So we take those documents and we put them together into a single corpus, and we process them using an NLP tool such as MetaMap. We can also restrict them to a specific vocabulary if we like. Next. And finally, what we end up with is this list of relevant concepts relevant to the phenotype in question that were mentioned in all of these knowledge base articles. And so in our case, concepts that appeared in at least 3 articles, we included in the dictionary. This dictionary being a representation of what concepts are important for defining COVID-19. Next slide. So now that we know more about how phi-norm works, we next ran phi-norm on data from both the UMC and KP Washington. We first created that AFEP dictionary of relevant COVID-19 terms by running NLP on 6 knowledge base articles. And we retained 159 relevant concepts that appeared in at least 3 of these articles. We selected index dates for when each patient was suspected to have COVID-19 based upon the date of first ICD code or other evidence. And we extracted all their notes within a plus or minus 30 days window of that index date and processed them using NLP to identify the concepts mentioned in the note. At KP Washington, they processed approximately 143,000 notes for a little over 8,000 patients. And at BUMC, we processed over 1 million notes for about 24,000 patients. The counts of NLP extracted concepts, the ones that were in that dictionary, were then transformed into patient feature vectors for input into the algorithm. And like I said, we process notes using MetaMap Lite, publicly available NLP tool commonly used from the National Library of Medicine. Next slide. So the other thing I've been mentioning is silver standard labels. These are labels that work as our best guess for whether or not a patient had the condition or not. And they're different from gold standard labels in that gold standard labels are usually created by expert review. They take a lot more time, but they're very accurate. Silver standard labels are kind of our best guess, and this is what we're giving to the algorithm, and it will essentially come up with a better decision than just looking at a silver label by itself. Instead of defining a single silver label, we used a few different ones to see how well the process worked as we investigated it. Our silver labels were the first one here, a structured label we're calling UO7.1 days. It's a count of the days with the UO7.1 diagnosis code. So UO7.1 being the COVID-19 diagnosis code. So how many days did they have that code in their chart? Silver label number 2 is a count of days with 6 COVID-19 related concepts or diagnosis codes. These included like diagnosis codes for COVID-19 pneumonia. And so we counted how many days they had those concepts. Structured label number, or silver label number 3 is an NLP label. It's actually just a count of how frequently COVID-19 was mentioned in the patient's chart. And NLP number 4, we implemented it slightly differently. at KP Washington and UMC. At KP Washington, we have the number of days in which COVID-19 concepts were found in patient charts. And at VUMC, we have the number of notes in which COVID-19 concepts were found in patient charts. Finally, we ran phenorm and evaluated the results. A brief note about that evaluation. So like I said before, phenorm doesn't require manually curated gold standard data, but we did need manually reviewed data to do the evaluation. Next slide. So we did manually review a random sample of notes from each site. Chart abstractors criteria is shown here. They not only identify COVID-19 infection status, but also COVID-19 severity based upon NIH criteria. On the left-hand side, you see evidence of COVID-19 infection, and they classify patients as either definite or highly probably infected, probable or possible infection, unlikely infection, not infected, or in some cases, insufficient information. The severity of illness scale ranges from asymptomatic to critical and includes common symptoms like fever, cough, sore throat, headache. Moderate symptoms, when we get into the moderate range, it starts to affect breathing, shortness of breath, dyspnea, abnormal chest imaging, and all the way up to respiratory failure and multiple organ dysfunction. Next slide. So in the end, we reviewed 483 patients from VUMC and 437 patients from KP Washington. And as I said earlier, COVID-19 can be asymptomatic, very serious, or anywhere in between. Using that criteria from our manual review, we actually defined two COVID-19 phenotype definitions. The first was at least moderate illness, showed here as moderate plus severity. So we wanted patients who had at least those moderate symptoms or worse. That was our first phenotype definition. Our second one was symptomatic COVID-19, mild plus severity, we called it here. So they had at least mild disease. This is opposed to like asymptomatic disease. We didn't attempt to identify asymptomatic COVID-19 because there wouldn't be any symptoms in the note to identify that. So we focused on these two here, moderate severity or mild / symptomatic severity. And for each one of those phenotype definitions, patients were classified as either a case, meaning they had that disease at that severity level or worse, or a non-case, meaning they did not have that. Next slide. So these are the results of running it on the moderate phenotype. And it didn't perform that well on the moderate phenotype, actually. So next. Yeah, so let's look at the first one here. So UL 7.1 days was our silver standard label. We said, okay, phenorm, here is our silver standard label, the counts of days with the UL 7.1 code. Here's all of the concepts we extracted from these patients' notes. Here's this AFEP dictionary list of important terms. And the results produced an AUC for the KP model at 0.7 and 0.814 at VUMC, which didn't sound bad at first, but if we look at the sensitivity of our model at 80% positive predictive value, it's not very good. So whenever we're saying 80% of the cases we classify as phenotype positive, they're correct. We're only capturing 29% of all the COVID-19 patients, all the phenotype positive patients in our set. Next, the 6 CUI days one, it's also poor, 50%, 47% sensitivity at 80% PPV for VUMC and only 5% at KP Washington. Next, the NLP concepts. These are the COVID mentions is the NLP based silver label that did not perform very well. Next. And again, we're still getting about 30% sensitivity for the VUMC one. So this is with our moderate plus phenotype, meaning that patients had to at least have moderate disease. Next slide. Now this one is better. So this is for symptomatic COVID-19, that mild plus phenotype. Next. And as you see here at the top, at both KP Washington and VUMC, the models both performed very well. We had a 90% AUC VUMC. The AUC for KP Washington was 0.773. And we both had a fairly high sensitivity at that 80% PPV threshold. So 99% at VUMC and 89% at KP Washington. Next. To highlight the next one here, those 6 CUI days. This is just a larger set of concepts that we use to define COVID-19. And again, VUMC, we're 95% sensitivity, KP Washington's at 88% sensitivity. Next. The NLP-based labels, KP Washington is doing very well with these, 98% sensitivity, identifying patients in this case. And one more time, and with the mentions or the CUI days and notes, these concepts found in these notes on different days or different notes, 98% for KP Washington and 95% for VUMC. In general, these AUCs are kind of close and may not be statistically different from one another, but you do see a trend of obvious improvement between the moderate or worse phenotype of COVID-19, and this one just looking at symptomatic COVID-19, which is much better. Next slide. So this is the moderate phenotype using silver label number 1, the days with the US 7.1 diagnosis code, VUMC in gold, KP Washington in blue. Next. So you can see here that 80% TPV in the VUMC data, we get a sensitivity of 35% when using that threshold of 0.52 that we identify from the model. Next. And for that one, that moderate plus phenotype, we see KP Washington didn't perform as well for that moderate phenotype. Next. Yeah, go ahead to the next slide. Another one here. So similarly for the moderate phenotype, we can see the results for that second silver label, the six concepts, 6 CUI days. Next, we get the 80% PPV, 47% sensitivity of VUMC, and only 5% at KP Washington. Next slide. For the mild phenotype, mild or symptomatic phenotype, using the US 7.1 shown here, as I showed the other slide, 99% sensitivity of VUMC at 80% PPV and 89% sensitivity at KP Washington. Next slide. And you can skip this one. This is the same data one more time. So the take-home messages on these slides. So relevance to Sentinel safety studies. Relatively modest effort was needed to implement this approach, and we replicated it in 2 heterogeneous settings, and it was fairly straightforward. It may be relevant for both chronic and acute health conditions. It's been shown to be relevant for chronic, and we showed it for an acute condition. The performance of automated models is affected by the fit between silver labels and phenotype definition, and the fit between source data and phenotype definition. So inpatient data might be needed to look at moderate plus severity. When the performance is less than desirable, automated approaches may still be useful starting point in model development too. So we've been using phenorm, but there's also PHECAP and MAP, multimodal automated phenotyping, and we're probably going to look into that as well. Next slide. This is described in more detail in an AMI abstract that we submitted, so hopefully that will be available soon. If you have any questions, please feel free to email me or David Carroll about this process. Thank you.
Thanks so much, Josh and David. It's really great to hear more about these methods and some of the early results of the applications, especially to the COVID-19 case in Sentinel. We're going to move on to our final speaker. And so, Adi, I'll turn it over to you.
Thank you. Today, I'd like to present some of the work I've been doing in large-scale natural language processing. Next, please. In particular, I'd like to focus on natural language processing that fit in this kind of desiderata. Obviously, we like to improve the phenotyping, performance measures on top of the structured data like ICD codes. We like to analyze large volume of clinical notes. No matter how large is your EHR, we'll like to be able to parse all the notes in the EHR to extract the relevant information. We like to have a data-driven generation of phenotype profile, and go into details in the next slide. But we also like to do a manual, minimize the amount of chart review. We like to have the generalization of the method across phenotypes, and also we like to have replicating results on other EHR repositories. Next, please. So this is the main architecture we have been developed, and I'd like to make the analogy of what we want to build here. It's a kind of a, information retrieval system like Google own electronic health records. So the next first component is basically when you type a query on Google, you kind of have an idea what you are looking for. So here we like to, build a phenotype profile or a phenotype query. However, we don't want to, to be kind of biased towards, towards the, people who type the query, and we like to use data-driven methodologies like unsupervised learning methodologies, such as word2vec to learn word embeddings that can give us ideas of a lexicon that are, words that are relevant for our phenotype query. Next, please. The second module is basically the information retrieval engine, which is basically, at the base of the core of this is the TF-IDF weighting measures, extracting, counts, document patient frequency, document counts, inverse document count, document frequencies, and also we like to apply on top of the negation of this phenotype expressions to be able to filter out those ones where those expressions are negated in nodes. Next, please. Then we like to refine our phenotype query with kind of a mechanism what is called query reformulation based on relevance feedback, to especially look at those polysemous word expressions that have some other meanings in addition to the relevance for the phenotype. Next. And finally, we like, the output of this kind of system is a ranking of the patients, and such that on top of the list will be the patients that are most relevant for the phenotype. Next, please. So we have been implemented over various phenotypes, this kind of system. We have published on this, started with extracting homelessness and their child experiences, published in JAMIA several years ago. And then we ported the code to our collaborators at Oregon Health and Science University. And we replicated the results for homelessness and also generalized the phenotype method to other phenotypes as well. Forgot to mention, but here occupation is another social determinant of health. And we recently have a paper that we published with our collaborators from New York University in Nature Medicine. And they were looking at the risk that the firefighters were exposed to World Trade Center disaster, like cancerogens and other environment exposure. And we helped them with phenotyping, applying these NLP methodologies to phenotype the controls, basically identify all the firefighters in Nashville and surrounding areas to use in controls in their studies. So this is kind of another application of what have been doing. And recently we applied this NLP methodology on suicide phenotypes, showing further the generalizability of the method. And in this current project, we basically look at the incidence of the suicide attempt phenotypes compared to all the other phenotypes above, where we look at the prevalence of the phenotype. Next, please. So basically, in the first component, these are kind of some examples that we extracted with word2vec, and so you can kind of have an idea of the phenotype profile. Next, please. These are the queries that we use for adverse child events and homelessness. Next, please. And these are some of the queries that we use for the suicide phenotype. This could be like more complicated to extract the phenotype profile. But I want like to make the difference. For example, to distinguish between suicidal ideation and attempt. For example, we use queries like thinking of ending his life, and then next please, and then compared to try to end his life, and so on. Next, please. And these are some of the some of the results we extracted for some of these phenotypes. And so we basically manually reviewed the top patients retrieved by these systems. And we basically wanted to prove that the top ones that are retrieved are the most relevant ones. Next, please. And we compare with the one we retrieve, we compare with the prevalence of their ICD codes for all these patients that we retrieve. So you can see here, you can have an idea what is the prevalence of ICD codes in all these patients that are retrieved, which is basically one of the characteristics of these phenotypes, that they are highly underreported and undercoded. And it basically shows that NLP also can help improve the coverage of extracting these patients. Next, please. And the idea is, so this is the output of our system. However, we want to extract and we want to use this, to see what are the, the cases and controls for various epidemiological studies. And basically the idea is, next please, is to use a threshold that say above this threshold, these are all cases, and below it's our non-cases. However, to compute this threshold, we need to, and we want to say, we want to extract these cases with a precision at least 70%, for example. But to compute this precision, you have to label all the cases. Next, please. And the idea is what we use is to use these NLP retrieve scores and the ranking of the patients and compute some probabilities to assign to each patient, such that the ones that are ranking on the top have a higher likelihood to be a case, and the one in the bottom, lower likelihood. Next, please. So basically, for example, if a patient has like a probability of 70%, and we do it multiple times, by sampling a uniform random variable from uniform standard distribution, we'll say like 70% of the cases will be like assign a case label. Next, please. And this is how we do it to be able to extract the precision at any rank in the retrieve list. Next, please. So this is the main idea, how we combine NLP by these probabilities and ICDs. So basically, we assign a higher probability, the one in the rank, and a probability of 0, one of the at the bottom of the rank, such that this probability is kind of monotonically decreasing. Next, please. Then we combine with the goal label such that, the ones that were already annotated will give them a probability of 1 to be a case. Next, please. And we also combine with the ICDs, since we also evaluate the ICDs, and we have, like, for example, for suicidal attempt, a very good precision of 86, I think. So we assign to all of them that have these particular ICDs, these kind of probabilities. So this is how we do a sampling and, do a kind of, a labeling of the cases. Next, please. And these are some of the results, and we're kind of trying to evaluate the negation detection by removing, all the patients that have this kind of mention negated in their notes. And we do find some improvement on that. And we also have wanted to compare what are the methodologies when you use only the probability rankings of NLP compared to the probabilities that use both NLP and ICDs. And these slides show basically that combining NLP and structured data, it improves the area under curves for retrieving suicidal ideation attempt. Next, please. And this is basically what the threshold that we use for extracting the cases. So above these thresholds, we extract the cases, and we're currently using a genetic study. Next, please. And this is basically what we're currently doing by looking at the, extracting the incidence of suicidal attempts. So a data point here is basically a patient and a day for this patient. For each data point, we want to see, so instead of, prevalence when you use only the patient, we want to extract the patient and the day here. And these are some preliminary results we extracted. Next, please. So in conclusion, I presented a scalable NLP system that we were able to generalize across different phenotypes, and we're also able to replicate the results across EHR repositories. We presented an unsupervised learning method for data-driven generation of phenotypes to extract query profiles. And we were focused on extracting the phenotypes cases with high precision. And we found that combining ICDs and NLP yield an optimal ascertainment of the phenotype. And we also demonstrated the feasibility of doing incidence of suicide attempt. Next. And with that, I'd like to thank you, all my collaborators that along the years have contributed to this. Thank you.
Thanks so much, Adi. this is really great to begin to hear some of the exciting applications that are happening and to see these tools be put into practice and in a multi-site way, which is really important within Sentinel. We have a few minutes for questions. And so I would like to, Adia, I'd like to start with you. Could you describe some of the challenges related to transporting your NLP system to running on other EHR repositories?
Yeah, this is really great. I mean, what I presented with collaboration with Oregon Health and Science University is the successful story, I should say, although I've been contacted by many other sites that wanted to apply our NLP system on their sites. However, there are some technologies difficulties when, because we rely on the scalability of the system that they have underlie on their EHR. And as you may know, there are different architecture of systems in different parts, and some of them may have different database infrastructures. I found that some of them even have nodes stored in archives, for example, older nodes, where you cannot be able to query. So we want all these basic computationals, like extracting the term frequency, document frequencies to be retrieved at scale in seconds from the entire EHR, and they were not able to do it. But some of the collaborators are able to do this, and we are able to replicate those. So there are some challenges, but it can be done for some of the EHRs.
Yeah, it sounds like some good initial steps forward, but big challenges there, and certainly relevant for Sentinel, who, we want to do this in a multi-site way. My next question is for Josh. Do you want to come back on screen? So you described the phenorm model, and you mentioned in your example that for identifying people with more severe COVID, it didn't perform as well. And so I was curious as to how generalizable you think that sort of finding is. Is this methodology, does it seem to you initially that this is going to be a methodology that's going to be better in identifying more general outcomes like any COVID or any diabetes, and more difficult with respect to disease severity, or is there something else happening in terms of the reasons for success or lack thereof for that case?
So phenorm is not necessarily applicable only to these general health conditions, but there are certain aspects of the method that make it easier to develop algorithms for general conditions. And for it to work well, we think that there are two conditions in particular that need to be met. The first one is that knowledge base articles that are specific to the health condition we're trying to model are really important, because those articles determine what terms go into the NLP dictionary and have a huge impact on what covariates or predictors are used. If there were knowledge-based articles specifically devoted to severe COVID-19 disease, then we would want to have used those, and we believe that the moderate plus or higher severity phenotype would have worked better. Second, the phenorm needs those silver standard labels to have a strong correspondence to the thing being modeled. And in all of our cases, the silver labels that we used were applicable to general COVID-19 or symptomatic COVID-19, but not very specific in terms of that severity. So that's something we're still working on.
Yeah, no, that makes sense. Relatedly, and this is just something I noticed as you were talking, so the sample sizes at the different sites were very different, with Vanderbilt having, big data, and the Kaiser site having, still large sample sizes, but relatively less. And these automated methods tend to be quite data hungry. Is that can you talk a little bit about the implications of that, and are there minimum thresholds that we need to be thinking about?
There are probably minimum thresholds. It did, as you saw, it worked very well at KP Washington for the symptomatic COVID-19. One of the things we did notice, though, is that VUMC has a higher severity group of patients than KP Washington, and we are inpatients, where you only had outpatients at KP Washington, and so we think that those qualities might affect the prediction in different ways.
Yeah, there's a lot of interesting heterogeneity to deal with in thinking about applying these methods. David, I will take one more question your way. You described a high sensitivity filtering, data-driven approach, and I was curious. what's the likelihood that even that high sensitivity approach might overlook some patients? And if so, what might be done to be able to recover overlooked patients in that instance? David, are you there, or are you on mute? We may have lost David, unfortunately. Well, let me turn to one more question before we go to the break, and this is for both of you. If you were to sort of, at this stage of the game with the work that you've done so far and what you know about Sentinel, what would you say is a key lesson that is important and relevant in thinking about the broader relevance of using NLP and even beyond Sentinel?
In general, I think that the NLP is important because something like 80% of information in EHRs is in unstructured form. We're still trying to identify how much of the important information is in that unstructured data, but methods like phenorm that automate this phenotyping approach, I think, are very important, and we should continue to test those out with different phenotypes.
Yeah, I would agree. I mean, NLP has a great role in many phenotypes, and especially from my experience, for those ones that have a low prevalence, that are underreported by structured data. And we have seen, and there are many phenotypes of these kind of characteristics where, for example, social determinants of health, they are very complex, and there are many of those, and unfortunately, they are underreported by structured data. And the only way to capture those and to improve the identification of those phenotypes is by natural language processing at this point.
Yeah, and we've certainly found that in our early work. I feel lucky to have had the opportunity to glimpse into this world and come alongside with some statistical knowledge of this area. So thanks so much to you both for sharing these examples today and talking more about these methods. I think next up is a short break, and so we'll let everyone step away for a few moments, and we'll be reconvening at the top of the hour. Thanks so much, you guys.
Thank you.
Thank you.
Hello, everyone. Welcome back from the break. I'm excited to introduce two speakers this afternoon. We'll be focusing on causal inference, and I'll start with Dr. Richard Weiss. Dr. Weiss is an assistant professor of medicine at Harvard Medical School and an associate epidemiologist in the Division of Pharmacoepidemiology and Pharmacoeconomics at Brigham and Women's Hospital. His research focuses on the development of analytic methods for causal inference and their application to large electronic healthcare databases for evaluating the comparative effectiveness and safety of pharmaceutical drugs. In this presentation, Richie will share ongoing work sponsored by the Sentinel Innovation Center to explore the application of natural language processing for large-scale feature generation and machine learning-based high-dimensional confounder selection for improved confounding control in comparative effectiveness and safety studies. So I'll turn it over to you, Richie.
All right, thanks, Shirley. All right, so in this project, as Shirley briefly mentioned, we're interested in better understanding how we can best leverage unstructured EHR data with the specific goal to improve confounding control in healthcare database studies. So next slide. Right, so just to give a very quick background, so confounding remains one of, if not the biggest challenges to being able to draw valid causal conclusions from healthcare database studies. Standard tools for confounding control within Sentinel have typically relied on adjusting for a limited number of investigator-specified variables. And while, of course, this works well in many applications, it can be limited in certain situations, like when we have new treatments that first come onto the market, the indication for treatment rapidly changing. It can be difficult to know all variables that we want to adjust for. But perhaps even the bigger challenge is, even if we know and can identify all variables that we want to adjust for, some of these variables are just not directly measured in routine care databases. Next slide. So this was an issue that was talked about in a paper published over a decade ago, where Schneeweiss and colleagues argue that we really should be thinking differently about how these healthcare databases capture confounder information. Instead of thinking of these databases as directly measuring information on variables that we want to adjust for, we can think of these databases as containing what they describe as a high-dimensional set of proxy factors that indirectly describe the health status of patients. A couple of examples that are given are frailty. In our studies, we almost always want to adjust for frailty or some concept of frailty, especially if we're conducting studies in elderly populations. But it's a health concept that's very difficult to capture in healthcare claims or EHR data. But instead, we could identify proxies for frailty that might be indirectly associated, like long-term use of an oxygen cylinder. And the idea being that if we can adjust for a very large number of these proxy variables, we could at least in part adjust away some of the confounding impact of these variables that are not directly measured. Another example is health-seeking behavior. It's very difficult to capture, but we could adjust for proxies like regular checkup visits or regular screening examinations. And so this is the idea of high-dimensional proxy confounder adjustment. Next slide. And so, this idea of high-dimensional proxy adjustment has become very influential in the pharmacoepidemiology literature over the last 10 years. This initial publication by Schneeweiss and colleagues, I think, has now around 1,000 citations, and there's been dozens of other papers that have been published that have built on this work to try to advance this idea of high-dimensional confounding control. For example, the OMOP collaboration, researchers like Patrick Ryan, Martin Schumi, who've been very influential with OMOP, have presented their own tools for this idea of high-dimensional proxy confounding control. And I think there's just really a growing literature that is showing that if we supplement investigator-specified variables with large numbers of empirically identified features using data-driven algorithms, more often than not, we can improve confounding control in our studies. But a limitation of current approaches is that they require data to be in a structured format, and they don't leverage information that's available in unstructured EHR data. So that's where this project wanted to explore more, is can we leverage unstructured EHR data to improve high-dimensional proxy confounding control? Next slide. So this project, the objectives of this project are related to, I believe it was Project Objective 3 that Rishi briefly talked about in the introduction, or Sebastian talked about in the introduction, where the overall Sentinel objectives of using NLP generated information from unstructured data. And so the application of NLP in this context is a little bit different from how we usually apply NLP or the other projects that apply NLP. Whereas when we typically apply natural language processing to unstructured EHR data, we're interested in identifying specific health concepts or phenotypes. In other words, we're very interested in interpreting the meaning of the generated features. In this context, though, we're simply interested in using NLP to generate very large numbers of structured features very quickly in a very automated, scalable way. And we're not interested in interpreting the clinical meaning of the generated features. All we want to do is generate structured features, which we can then throw into machine learning algorithms, which can evaluate empirical associations in the data to see if there's any confounder information that is captured in those generated features. So the context is a little bit different here of how we're applying NLP. So it's really divided up into two different phases. In phase one or aim one, we use scalable application of NLP. We look at different approaches to generate structured features from high dimensional EHR data for proxy adjustment. So this first aim, this is actually quite a large undertaking. It wasn't possible to do this within the time constraints of the Sentinel project. And so we were able, though, to leverage work that's been done with an ongoing R01. It's being led by Josh Lin, who's the PI, and myself and Sebastian Schneeweiss are co-investigators. This is a typo here, not co-PIs, but co-investigators. But luckily, we're able to leverage work from this R01, which in this R01, we applied the NLP tools to generate the structured features. And then in aim 2, which is what this Sentinel project focuses on, once we have this generated data, which is what's sometimes referred to as ultra high dimensional data, how can we best apply machine learning tools to optimize confounding control with this information to improve proxy adjustment? And so that's what I'll be talking about here. Next slide. So the data that we're using is EHR data from Mass General Brigham. This is Mass General Brigham's the largest healthcare provider in Massachusetts. We identified patients 65 years and older, and we were able to link some of these individuals to Medicare claims data. I think in the end, we ended up with around 1 million individuals where we were able to link with their claims data. With this linked EHR claims, we generated 3 cohort studies across different therapeutic areas. And the purpose here is we have these studies as case studies where we can evaluate and test various methods where we've generated NLP features from unstructured EHR data, and then we can test different methods for ultra high dimensional confounding control. So ultra high dimensional confounding control, that's the term that's often used in the biostatistics and epidemiology literature, just meaning that the number of features is much larger than the sample size. Next slide. So to generate structured features from EHR, we used a very simple approach called bag of words. So this is a very common flexible NLP approach. And so the advantage, of course, is that it's very fast, simple, scalable. The disadvantage, though, of course, is we potentially lose information because of its simplicity. What it's doing is it's just measuring the frequency or occurrence of the words within a document. And so the order and the structure of the words within documents is discarded, potentially losing information. We're only concerned with whether or not the word occurs and not how it relates to other words. Of course, there's other NLP tools that could potentially be applied, which might be future work. But to begin with, we started out with the simplest approach of bag of words, and we can move up from there. Next slide. So this is what our cohorts look like. So after we linked the data and then generated structured features from the unstructured EHR, this is the sample size and variables that we're dealing with. So I'll just point a couple things out in this table. If we just focus on empirical example 1, the first row, we see that we have a study population of about 3,500 individuals. So this is a much smaller sample size than what we would have had if we were using claims data alone. So one of the challenges when we're utilizing linked EHR claims data is that oftentimes it reduces our sample size, sometimes quite substantially. But an advantage or potential advantage that we want to explore here is that while we reduce the sample size, we also have much more information to potentially improve confounding control. So we have 39 investigator specified variables, 18,000 claims codes. And so this information, the investigator specified variables and claims codes, is what we traditionally have in pharmacoepi studies, where the claims codes could be used for different algorithms for proxy confounder adjustment. So the unique or difference here is that we have this additional EHR features, where we use bag of words to generate about 20,000 structured features from unstructured EHR text. And so we have this ultra high dimensional data where the number of features just greatly is much greater than the study population, and how best to analyze this data to maximize confounding control is what we wanted to explore. Next slide. So we began by considering various methods for confounder selection and propensity score estimation. We focused on lasso-based approaches. We did this for a couple of reasons. Lasso by far is the most popular or widely used approach for high dimensional confounding control in the biostats and epidemiology literature. And in the pharmacoepidemiology literature in particular, lasso has been shown to perform particularly well. It's the approach recommended by OMOP. I think a large reason for this is with pharmacoepi studies, the type of data that we're dealing with, we often have very large numbers of dichotomous binary variables. And so the parametric assumptions made by lasso actually work quite well in these settings. But we looked at different variations of lasso. We looked at the regular lasso model, which simply fits a lasso model to optimize predictive performance. And then we looked at 3 variations, which fit a lasso model focused more on optimizing the properties of causal effect estimates. We looked at an outcome adaptive lasso, which was developed by Susan Shortreed and her colleagues at, I believe it was University of Washington, the biostatistics group out there. We have the collaborative controlled lasso developed by Mark Vanderland and his team at UC Berkeley with their targeted learning tools. And then this outcome highly adaptive lasso, which is just a combination of the outcome adaptive lasso and the collaborative controlled lasso. Next slide. So initially, we were just going to apply these tools directly to the 3 cohort studies that we generated. We generated those cohort studies specifically because we had a general idea of what we thought the treatment effect might be based on clinical trials and other evidence. But after getting into this project, myself and the collaborators, colleagues on this project, we decided that that's not the best idea because while we might have a general idea of what the truth might be, we still don't know exactly what the truth is. And so if we're going to make recommendations on the performance of these methods, we need more of an objective approach to evaluate performance and make recommendations. Instead, we used what are sometimes called synthetic control studies, which is just a fancy term for simulations, simulations that are developed in a way that try to reflect the complexities of the real world data as best as possible. There's many publications within recent years of different ways of generating synthetic control studies. The details really aren't important, but that's the approach that we took in this paper, is to generate simulation studies, synthetic control studies to evaluate performance and make recommendations based on those, and then afterwards apply the tools in the actual empirical studies. Right, next. So these are the methods that we evaluated. So again, these are just different variations of lasso. We have on the first row the standard lasso model, which these are all lasso models to fit the propensity score model. And so which combines the confounder selection and propensity score estimation just into one step. So we have the standard lasso, which is just a traditional lasso model that fits the model or selects the penalty factor with the goal to optimize treatment prediction or cross-validated treatment prediction. And then we looked at various alternatives to the standard lasso, which fit a lasso model with the goal more for causal inference. We looked at 2 variations of CTMLE lasso, which stands for collaborative controlled maximum likelihood estimation lasso. The difference between these variations of the CTMLE lasso are not really important, so I'll just skip over that. But what the CTMLE lasso does is, instead of choosing a penalty factor to optimize treatment prediction, it chooses a penalty factor to identify variables that have the greatest impact in the fluctuation of the treatment effect. In other words, it's trying to identify variables that have the strongest confounding impact rather than just trying to find the variables that have the strongest effect on treatment. The outcome adaptive lasso is another variation that fits a lasso model for more of a goal of causal inference, where the outcome adaptive lasso, again, developed by Susan Shortreed and her team out at University of Washington. It fits a lasso model where it puts heavier weight on variables that are weakly related to the outcome. So it tries to exclude those variables from the model. And then the bottom two models here, CTMLE, OAL, and these are a combination of the CTMLE lasso with the outcome adaptive lasso. And the difference between those two variations, those two bottom rows, is not really important for this presentation, so I'll just ignore those. We can maybe circle back if there's time at the end. Next. So yeah, next slide. So I'll just share a couple of, so these are just some selected results from some of the simulations we're seeing that I think kind of tell the pattern of what we're seeing of what works well in this type of data that we're dealing with. And so this figure shows the predictive performance of each of the methods. The left panel shows the predictive performance in the same sample in which the model was fit. So this is a measure of how much overfitting the model does, whereas the right panel shows the cross-validated prediction performance. And so for discussion here, we'll just focus on the right panel, as this is actually the more important or relevant information, the cross-validated predictive performance. So on the y-axis, we have the cross-validated C statistic, and on the x-axis is different scenarios, different simulation scenarios, where for each of these scenarios, we held constant the sample size, the study sample size of 5,000, and we also held constant the number of confounding variables that we simulated, which was 100. And what we varied was the number of baseline variables that were spurious variables, or just noise that we threw into the data. So on the left-hand side, we had 100 confounders, a sample size of 5,000, and then 1,000 spurious variables, whereas on the very far right, we have 100 confounders, a sample size of 5,000, but then 10,000 spurious variables, so much more noise that we're throwing into the model. And the idea here is we want to better understand how these models perform as the noise in the data increases, because as we get into this ultra high dimensional data, that's what's going to happen, is we might get additional confounder information, but there's also going to be a lot more noise that we're throwing in as well. So in terms of predictive performance, we see that the method one performs best. The method one was doing the best job of being able to identify important predictors of treatment in this very noisy data. If we just look at the very far right points under the on the x-axis under 10,000 or 10K, the red triangle, which is method one, just the traditional lasso for optimizing treatment prediction performs best in terms of cross-validated predictive performance, which is not surprising. There's a little bit of a cost in terms of predictive performance when we try to fit a lasso model to optimize performance for causal inference. So next slide. But if we looked at results for bias, we see something that's a little bit more interesting. So again, just focusing on the very far right results under the scenario where we have 10,000 spurious variables. So this is, again, the simulation scenario where we have a large amount of noise that we're throwing into the data set. And we want to know which one of these methods is best able to sift through all of that noise and identify relevant confounder information. On the y-axis, we just have the absolute bias averaged across all of the simulation runs. And these are just selected results. We looked at additional scenarios as well, but what we consistently saw is that when we combined properties of targeted learning, so model 6 here, where we combine the CTMLE with the ideas of the outcome adaptive lasso, we consistently tended to perform better in terms of bias reduction versus just the traditional lasso, which is currently recommended in the literature for this ultra high dimensional data. Next slide. So just some general points for discussion. So we found that selecting models when we have this ultra high dimensional data where we're generating features, structured features from unstructured EHR text, where the number of features is much greater than the sample size. Selecting models based on collaborative learning tended to improve bias reduction, even though predictive performance declined. The outcome adaptive lasso combined with principles of collaborative learning generally performed best. Something that was interesting that we found was that some degree of overfitting these models that did perform best in terms of bias reduction tended to overfit the model, select more variables than the traditional lasso model that was just trying to optimize treatment prediction. So some degree of overfitting was beneficial for confounding control in this type of data that we're dealing with. One other thing that we noticed, though, is that bias increased as the number of spurious variables increased, which is not too surprising. We found that the bias resulted from two sources. One is that the lasso models or the methods not selecting the confounding variables. It became much more difficult for each of these methods to identify the relevant confounder information as the noise in the data increased. But there was also this bias that was due to regularization bias from the lasso model. And this has been talked about in the literature. So to deal with this second type of bias, this regularization bias, we looked at variations of the lasso called the relaxed lasso. I think I'm going to skip over this for now because I'm about at the end of my time. But essentially what the relaxed lasso does is just fits the lasso model twice, where we first fit the lasso model to select variables, but then we fit the lasso model again to reduce that regularization bias with less penalization. Again, the details really aren't that important, but this is something that we're currently exploring with the project that's ongoing. And so, yeah, I'll just skip over the details of the relaxed lasso. So you can skip these results and just go to my concluding slide. Yeah, again, so we found, yeah, so just go back to future work. Yeah, that's great. So yeah, so the future work next steps is we think we've got a pretty good idea for the patterns of, what methods tend to work well, at least in these, simulated data sets where everything is tightly controlled. There are some complications of how well do these simulated data sets reflect the actual empirical studies, and that's something that we're currently discussing and working on with the group. If there's time, we can talk more about that. That's something we can discuss here, but at least within the simulations, we have a pretty good idea of what works well. And so the next step is to apply these in the actual empirical studies and see kind of what results and patterns we see. And so the next step, I just need to acknowledge all the, or the next slide, sorry. Yeah, so just acknowledge the team, which has been just really helpful with this project. We're applying a lot of complicated methods here. So the insights from, we have a lot of really talented biostatisticians from FDA, Berkeley, Jen Nelson at Kaiser Washington, University of Washington, University of Michigan, just a great team here that's been super helpful with this project. And so with that, I'll just, if there's questions, happy to answer any questions.
Great, thank you, Richie. I'll get the ball rolling with a question. So the first step for high dimensional proxy adjustment is generating features which you can use for confounding control. And you've focused in this project on a relatively simple NLP approach, the bag of words approach, which you said was for simplicity and scalability. Have you considered other NLP approaches that could scale for the objectives that are described here?
Yeah, so that's the next natural step in this work, which I don't know. I mean, we're not going to be able to tackle that in this current Sentinel project, but along this lines of work, that would be the next natural step. Is to the biggest, I think the biggest challenge is trying to figure out how best to generate proxy variables from the unstructured EHR data, and that could be a whole area of research unto itself. Here again, for simplicity, we just started out with the simplest approach, bag of words, but there's many other approaches, distributional semantic models, word embeddings, that consider, relationship with other words, the relationship between sentences, that, in theory could do a much better job of generating proxy variables that can capture meaningful information, confounder information. There's always, for the purposes of high dimensional proxy confounder adjustment, though, there's this scalability issue. So there's this trade-off between capturing information versus scalability. And so how far you can go in the complexity of the NLP tools is really unclear at this point. And so that's something to explore in future research.
Thanks. There was a question in the chat, which I think you sort of addressed, but maybe you can say a few more words about it. Well, the question was, if bag of words is used, do we need to be concerned about irrelevant adjustment?
Yeah, so that's always a concern for high dimensional confounding control in general, is over adjustment or unnecessary adjustment. So, and that's where the tools for these lasso-based extension tools come into play, where we generate these features, but then we throw all of these features into these machine learning tools for causal inference, for confounder selection, to try to tease out what variables are important confounder information. It's a really challenging issue. No tool is perfect, and it's almost certain that in any practical setting, there is going to be some unnecessary adjustment going on. The goal, though, is that the the downsides of adjusting for some variables unnecessarily are outweighed by the gains of adjusting for additional confounding information. And there's some good papers on this that talk about the problems of adjusting for things like instrumental variables and colliders. But I think the evidence, more often than not in every scenario, but more often than not, the evidence tends to show that it's better to err on the inclusion of variables at the expense of adjusting for instruments and colliders. The bias caused by adjusting for colliders and instruments tends to be smaller than the bias caused by not adjusting for additional proxy information that potentially contains confounder information.
Thanks. I think we have perhaps time for just one more question. You alluded to some remaining challenges that you have with the empirical studies above the simulation. Do you have time to say just a few words about that?
Sure. So yeah, so this is an area of ongoing research and debate as well. So when we have all these decisions for causal inference, the question is how do we best tailor our analytic decisions to the study at hand? Well, there's a lot of papers that have been coming out recently that the way to do this is to build simulation studies that are as realistic as possible to reflect the properties of the actual study population. And so there's different ways of doing this that I mentioned. No approach is perfect, and we can never perfectly replicate the complexities of the actual study population. And so when we apply our tools to the simulation studies and we see certain patterns, we can never be certain that those are actually going to be the best methods for the actual empirical data. And we actually saw that a little bit initially in the examples we saw in this project. And so we applied our tools to our simulation studies. We saw this pattern that collaborative controlled outcome adaptive lasso models performed much better. When we applied them to the actual empirical data, we saw results that were a little bit unexpected. And we think that what's going on is that there's some hidden bias in the data that we weren't able to replicate within the simulation studies that are making results look strange. And so the question of how can we best tailor analytic choices for causal inference to the actual empirical study is a real challenging issue, an area of ongoing research and debate. And we're grappling with that here in this project as well.
I think the outputs of this project will become very valuable for future researchers who want to use that sort of framework. Well, thank you, Richie. So I guess we can move to our second presentation for this afternoon. I'm delighted to reintroduce you to Dr. Desai. He'll be giving the second presentation focused on causal inference. Dr. Desai is an assistant professor of medicine, also in the Division of Pharmacoepidemiology at Brigham and Women's Hospital and Harvard Medical School. His applied work focuses on chronic disease pharmacoepidemiology, and his methodologic research is aimed at improving inference from observational studies. Rishi, as you know, is also the executive director and operations chief for the FDA Sentinel Innovation Center. So turning it over to you, Rishi.
Great, thank you, Shirley, for the very kind introduction, and thank you everybody for staying until the very end of this 3-hour session. So this is a work group where we are developing a causal inference framework for Sentinel projects, for future Sentinel projects. But I think the principles that we are developing are also quite applicable to studies conducted using routinely collected healthcare data more generally in the field. So the first slide I have is around the motivation for doing this work. So as you may be familiar, there are plenty of tools and guidelines already available in the literature, and you may have seen some of these already. For example, we have something called a Robbins, we have a record PE, we have start RWE. So a natural question is that why do you need a new framework? Why do you need new principles to do a study in a proper way, in a way that is replicable and transparent? And the way we were looking at it is there are guidance documents, or there are various tools for various purposes. So if you kind of group these existing tools, what you have is a set of tools that are strictly for quality assessment. So if you're familiar with Robbins, it's really a tool meant to assess the risk of bias in non-randomized studies of intervention. So basically what you have is a checklist of things that you can take and look at a paper that is published or a study that's conducted and see what is the risk of bias. And this is really developed for meta-analysis and ranking studies in terms of bias for meta-analysis. So these are, GRACE is a similar concept. So these are quality assessment tools. There are tools for reporting, improvement of reporting. So START RWE is the most recent example, which is a really nicely thought out example of how to think about various steps in reporting when you are conducting or writing a protocol for your study. You have guidance documents from these regulators. For example, FDA put out something very recently. EMA has its own version, the NCEP pharmacoepidemiologic methods standards. These are best practice documents. They are almost like a textbook where you have all the information there. It's not a framework where you have various steps along the way, which will help you guide your decisions. But these are really all-encompassing frameworks or guidance documents, almost like textbooks. And then you have specific, highly specific versions. So, for example, OMOP or Odyssey has something called a legend, which is highly specific to how they do the studies. And so the way we are thinking about this is that we have various tools in the literature for quality assessment, for reporting, for describing the best practices. What we don't have is that none of these tools offer a general framework to guide decision-making at various steps along the way. And so we came up with, along with the FDA, came up with this vision for a framework to guide principal investigations using non-randomized secondary data. And this is actually quite important because if we have a stepwise process that systematically considers key choices when you are thinking about design of a study and analysis of a study, then you can generate more confidence in the results that you eventually end up with if you are following a systematic process, if the process is carefully thought through and it's not something that you make up as you go. And so that's the exact reason why we kind of gather this work group to develop a standardized or industrial process which will serve as a guide to inform conduct of non-randomized secondary database studies for drug outcome evaluation. And for specifically for Sentinel program or for the FDA, the key considerations here are that we have to limit variations across investigators by outlining a general process. And this is important because as new queries come in from the FDA for the Sentinel program, it is inevitable that no same group or same investigator is implementing these queries. And when that happens, it is likely that the different groups of investigators may make decisions slightly differently based on how they approach that same question. And so we want to minimize that type of variation by providing a map or a roadmap of how to kind of systematically think about each and every decision. And so focus is on repeatability of the process. If we do that, we can generate evidence that inspires more confidence, because regardless of who is implementing these steps, if these steps are nicely outlined, if all the considerations are nicely outlined, no matter who designs your study, it is likely to be quite homogeneous across groups. And so that's one of the key motivations behind developing this new framework. And it's also important for us to have it written and endorsed by independent experts who have no stakes in any of these studies, no financial or any intellectual kind of conflict of interest when writing this. So it's an important step forward, we think, for the Sentinel program, but the lessons that we have are more general. And so what I have for you today is a draft of this proposed framework. We don't have any data or anything like that. It's a stepwise guide or a proposal for various steps along the way as we are thinking about implementing new studies for drug outcome evaluations. And so we basically are thinking of this as a series of steps. The first obvious step is to define a well-defined research question in a target trial framework. So target trial framework is something that has gained a lot of popularity over the last 5 to 10 years, and it thinks of all observational studies or all non-interventional studies as an attempt to emulate a hypothetical randomized controlled trial, a prospective study. So we think that this is a really important and valuable mechanism to outline your research question in a principled way. And we also recommend leveraging the PCORD framework. I'll talk about this in a bit as well, but this is where everything starts. So the step one is a well-defined research question. Then you move on to step two, which is dealing with the key question of what are the data sources that you require to address the question that you just outlined in your step one. And for each of these steps, I have separate figures, so I will go into those a bit more. First, I'll introduce the overall framework, but that's our second step. It's the fitness for purpose. How do you define that? The step three is then to refine your target trial parameters. So you have your target trial specified in your step one, but then you need to translate all these kind of concepts into your study parameters, and you need to outline them quite clearly. So that's your step three, basically a protocol development type step. From there, you move on to what is known as a feasibility assessment, or a preliminary look at your data to see what you are proposing to do is feasible based on the data source that you have access to. And we also have stepwise guidance in how to do that. And this is something I think is at the moment is lacking in the literature, where you don't really have a good sense of how to approach feasibility analysis without kind of violating this outcome blinding principle, without getting into kind of the analytic phases. And so we have a nicely thought out stepwise guidance for that step as well. And so once you have the feasibility assessment, then you may actually go back to refine your trial parameters, target trial parameters further, based on either finding out that the outcome events, for example, are not sufficient, not feasible to support your study. Then you may want to go back and relax some of the criteria that you make in step three. But in other cases, you may also find out that the comparison group, for example, that is selected is actually in clinical practice not being used interchangeably with the exposure group of interest. In that case, you may want to go back again and fix your or select a different study population, for example. So it is kind of a loop without going into the analysis phase. Just based on feasibility counts, you may be able to kind of refine your study protocol. So once you have these 4 steps worked out, you may want to go on to pre-specify all your robustness evaluations. So these are all your sensitivity analysis, and we again have a framework for what to consider there as well. And once you have all these 5 steps outlined, then you may want to move on, may be ready to move on to the final inferential analysis step. And I want to again highlight that steps 1 through 5 are all considered study planning steps, where you are not really looking at any data in terms of addressing the question that you posed. This is all in the planning phase, and you are only analyzing your data in the step 6 when everything else is pre-specified, which is your inference step. So this is just an outline for all the 6 steps that we have proposed. I'll go over quickly one by one all of these steps that we briefly looked at. So this is the most straightforward step, where you need to define a research question in the target trial framework, and a lot has been written mostly by Miguel Hernan and Jamie Robbins. And we firmly endorse that, because we think that this is a non-negotiable step in any framework. You need to be able to articulate what's the question that you are attempting to address. So this includes specifying the patient population of interest, as in P, intervention, comparison, outcome, along with the time horizon that you are interested in, and the setting in which the study is implemented. So this is not very complicated. I think we can all agree to all these principles. From there, you move on to your step 2, which is the fitness for purpose of data sources. And we focused a lot on insurance claims data because, as you may know, Sentinel's backbone is insurance claims. Sentinel has a large network of insurance claims enrollees, which is used for most of the queries that are run in the system today. So you start with addressing a few basic questions to determine whether the insurance claims are fit for purpose or not. The first most important question is whether the exposure of interest is captured in your data source or not using insurance claims. For most cases, if the exposure is something like a prescription medication, the answer will be yes, and then you can stay within that framework or within that data source. If no, if you are interested in something like blood transfusion products, for example, you may need to look for alternate data sources, which is my second box where we are thinking of additional sources. So this is the first kind of branching point. Then the other two important questions are whether the outcome of interest is captured in your insurance claims with sufficient validity, and sufficient validity is based on measures like PPVs, for example, depending on how common or rare your exposure is. And similarly, you also want to make sure that the population identification is appropriately available in insurance claims or not. And in some cases, if your outcome or population identifier is a serious event like myocardial infarction, you may be just fine with insurance claims. But in other cases, if this is something more nuanced, like pancreatitis, which is an example that David also used in his talk, then you may need linkage to EHRs because insurance claims may not be sufficient to address or capture this outcome of interest. Similarly, for population identification, if you are thinking of something like something more nuanced diagnosis like heart failure with preserved ejection fraction, for example, then you may not be able to do justice to that kind of nuanced diagnosis just using insurance claims. You may need linkage to EHRs. So once you have this linkage established, what you can do is you can develop and validate these claims-based algorithms. And if those turn out to be performing fine, then you may want to take it back to the larger, broader network of insurance claims to leverage that. But in cases where you are just not able to do well with just claims-based parameters based on these validations, you may want to restrict your study population to those with linkage. And finally, the fourth key question is around the identifiability of confounding variables. So this is kind of lower on the list because it's in many cases it's nice to have or great to have, but may not be absolute must. And so if there are plenty of diagnosis for indications or medications are sufficient for confounding adjustment for your purpose, then you may be able to stay within insurance claims. But in some cases, lab results may be highly predictive of treatment as well as the outcome. For example, renal function is a common one that comes up. Then you again need linkage with EHR. And once you have linkage, again, you have a bunch of options, but one common option would be to derive additional information on unmeasured confounders that are unmeasured in the insurance claims and take that back to the insurance claims for additional confounding adjustment or calibration type of analysis. And then based on how you address or answer these questions for your specific research area, you may be able to decide what data sources are fit for purpose for your question. And so this is again a framework for an overall thinking process of how to think about fitness for purpose of data source based on 4 key elements of data capture, really. So this is our second step. From there, you can go on to your third step where you have now a research question ready. You roughly know how what data sources you're going to use. Now you can start specifying or hashing out more details on what you want to study in terms of a specific protocol. And this is again in a target trial framework. Again, much has been written by folks on this. So this table may look familiar to you. And here you may want to first start out by saying, okay, if this is my research question, how would I do this in a prospective way, in a prospective randomized controlled trial way? And then you start writing that out here, and then you can translate it to how you are able to kind of mimic some of these principles using the data that you have at hand. So for example, in medication-based studies, random allocation would be ideal in a prospective setting. However, what we have instead is prescription dispensing data, and so we may be able to use that to kind of mimic the initiation of medication in the treatment setting. And so there are various considerations, and again, some of this can be quite technical, so I don't want to get into too many details here. But I just wanted to highlight that the purpose of a table like this would be to be explicit with all your assumptions and all your design decisions that you make, and have it out there for people to look at in your planning phase. So once you have these 3 steps nailed down, then you can actually go on to do some feasibility analysis. And this is kind of a thorny issue, because when you talk about feasibility analysis, you basically are touching your data at this point. And we think that there are principal ways of doing this, and this is our attempt to kind of highlight or outline what these ways are. So we think of feasibility analysis in various steps. So the first basic step is the cohort feasibility. And this is very important for a program like Sentinel, because in many cases, the research question of our interest is around new medications. And so when you have a medication that is just approved, you may want to see if that is used frequently or not before moving forward. And if it is, if cohort size is feasible, then you may move on. But if not, then you may want to wait until more data is accumulated. So once you have the cohort size, you basically can have 2 parallel paths. One is diagnostic evaluations. The other one is outcome feasibility. Outcome feasibility is just looking at, without stratifying by the treatment group, just looking at the rates of the outcome in just the reference group, for example, or the combined reference and exposure group. You see if the outcomes are occurring at a sufficient rate for you to do a sufficiently powered analysis for the question of your interest. So that's straightforward. The diagnostic evaluation is something more nuanced, where we think that it's useful or important for you to look at key patient characteristics to see if you are enrolling or recruiting a cohort that may be reflective of who you want to study. You may also want to fit your propensity score model to see whether you are able to achieve balance within your control and reference control at exposed group, and may want to fine tune that model at this stage, at the feasibility stage, to make sure that you have sufficient overlap and whatnot. And so if the diagnostics are passed, if outcome counts are great, then you can proceed to step 5. But in case where there are potential issues that are diagnosed in either of these feasibility paths, you may want to consider going back to your step 3, which is your study protocol, and make fine-tuned adjustments to your design to either get back to a level where you may have a feasible study, or you may want to decide that this may not be the right time to do this study, and you may decide to wait. And so this is a principled approach to do the feasibility analysis in our view, and this is what we have outlined so far in this framework. Once you have step 1 through 4, you can move on to pre-specification of robustness evaluations, and this come in various flavors. I will not spend too much time, but just these are all kind of deterministic sensitivity analysis, which is something that we routinely employ, where we vary one design parameter at a time, and then see what happens to our parameters, to our result parameters. There are quantitative bias approaches, which is more, in some cases, maybe more probabilistic. And there are net bias analysis, which includes things like negative control exposure or trial calibration approach where you may want to see if you are able to recapture the effectiveness outcome that is published by a trial to have more confidence in your ability to study other outcomes which are not studied in this trial. So these are kind of robustness evaluation. We think that this should be a standard part of a study planning phase and should be included at the planning stage. And so these are our 5 steps we think every investigator should be able to follow before moving on to the analysis phase of a study. So this is my brief summary of the outline that we have developed for this causal inference framework. We are continuing to fine tune based on engagement with various stakeholders. We are conducting also a demonstration project to highlight how decisions are made when you're faced with these questions in a real case example. And finally, the goal is to disseminate this by early next year in a peer-reviewed publication for the Sentinel program to have a framework that they can adhere to, and hopefully this will be useful for the public or for the scientific community at large. So that's my last slide. I will stop here for any questions or any comments that you may have.
Great, thank you, Rishi. Sorry, we have 5 minutes for questions, and I'll start with one. You spoke at the beginning about how this framework fits in with the overall landscape of other existing tools, frameworks, and guidances. But could you elaborate a little bit more specifically on the relatively new ICH E9 guidance on clearly defining estimands, and also the Harper protocol template for real-world evidence studies?
Yeah, so that's a very good question. I think for us, clearly there are aspects that we are touching which other tools or frameworks are not kind of tailored to do. So in that way, we are kind of additive, but we also want to make sure that this is complementary, and the ICH estimand principle for clinical trials is something that is quite relevant here, because some of the terminology that we use versus what's in there is different, but concepts are very similar. And so our approach or our hope is to kind of draw parallels whenever possible with existing guidances or existing tools, to say that we are generally mostly in agreement or endorsing these principles that exist, but this is highly specific to the type of questions or nuanced consideration that the Sentinel program faces on a routine basis. Similarly, the other example would be a protocol document, for example, the START RWE, or the new update or a newer version that the ISPOR-ISP task force led by you and some others are working on. So we are very aware of the environment and other existing tools. And we are actually using this ISPOR-ISP, for example, the ISPOR-ISP protocol template to see how the principles that we are endorsing translate to these types of documents, to make sure that these investigators don't have to kind of do that kind of translation, and we kind of provide them provide the readers with an understanding of how these documents are complementary and build on one another.
Thanks. Seeing no questions in the chat, I'll ask one more before we close out. You spoke a little bit about the types of questions that would be asked for Sentinel. Can you speak to the flexibility of the framework to accommodate different types of research questions?
Yeah, that's a very good question. We have tried to keep this more general. Although, as you may know, Sentinel deals mostly with drug safety questions. So that is our kind of bread and butter type studies that we have had in mind when talking about most of these studies. But if you look at our principles, they are quite general in terms of if you have a study a comparative study that is focused on other types of outcomes, like patient-reported outcomes, for example, or effectiveness outcomes, these principles would nicely kind of be able to accommodate those types of questions as well. So we see this framework as a really general guide to do principled studies of any outcomes associated with treatments using non-interventional secondary data sources.
Wonderful, thank you. I see no more questions in the Q&A, so I'll hand it back to you for closing remarks.
Okay, great. So thank you again, everyone, for joining us. As we have seen, the Sentinel Innovation Center has been quite busy with various projects in our portfolio. We are making strides in helping to achieve the FDA's vision of a medical data enterprise with a query-ready system containing more than 10 million EHR lives. That is briefly introduced by Sebastian at the beginning of this talk. Through projects that we have undertaken in key research needs, which is really grouped into these 4 strategic priority areas, and we have heard a little bit about the initiatives in each of these areas of infrastructure building, feature engineering, causal inference, detection analytics. We are making steady progress in identifying salient challenges that are presented by EHRs. Of course, everybody is quite excited and really hopeful about the value that EHRs are going to add in improving the quality of the studies, but we are also equally kind of aware of the challenges that this is going to bring. For example, the unique mapping challenges that Keith described. So these are really unique to EHR data sources, which require careful planning and careful thinking in tool building, especially when we are thinking about a program like Sentinel, which has really regulatory implications once the studies are conducted and out there. And so we are thinking about these proactively and trying to attempt those to the best of our abilities through initiatives like the ones that you heard about today. And through this highly interdisciplinary work, which is conducted all around the country, really at great institutions, with involvement from various fields, experts from various fields, including epidemiology, informatics, medicine, pharmacy. We hope to generate unique insights regarding meaningful use of EHRs for clinical research, and hopefully provide some practical solutions for people to implement. So this is our hope, and we also have outlined our vision in this manuscript, which was recently published, which you can feel free to look at. And with that, I really want to thank all the participating organizations of the Innovation Center, which is listed on this slide, and there are many of them. And I again want to thank you for staying until the end today. And if there are any comments or questions for us, any suggestions, any exciting tools or methods you're working on that you think could benefit the Sentinel program, we would love to hear from you. The plan for Innovation Day would be to do something similar with more projects and more updates every year. And so we look forward to seeing you again in 2023. And so with that, I think we can close this session. Thanking again everyone for your time today, and thank all the speakers for their wonderful presentations.